Scientific Affairs in Institute of Dendrology, Polish Academy of Science. Welcome at the International Conference Research and Practice in Forest Ecology. For the smooth contact of the conference and organization issues, please refer to the organizing committee led by Dr. Karolina Sobierajska. Dr. Sobierajska will be providing you with all the necessary information during the conference. Now, I would like Professor Andrzej Jagodziński, invite the director of the Institute to officially open the conference.
Good morning for everyone. I think that I am as stressed as you. You have just come to Kórnik. I hope that the trip to Kórnik, the journey to Kórnik was very funny and the way was really very good to meet with us. Before I will start my presentation about our institute, I would like to warmly welcome, just a second please. Dzień dobry, zapraszamy serdecznie. Bardzo proszę. Okay, so now we are all of us. I warmly welcome you to the International Scientific Conference Research and Practice in Forest Ecology, organized by the Institute of Dendrology of the Polish Academy of Sciences. The conference, as you know, is organized within the framework of the Excellence Science program announced by the Ministry of Science and Higher Education, and I would like, right now, to kindly thank for the financial support of the Ministry of Science and Higher Education, as well as additional funds received from the Zakłady Kórnickie Foundation. Thank you very much. And now please let me allow to welcome our guests. I would like to kindly welcome the President of the Zakłady Kórnickie Foundation, Dr. Dariusz Grzybek. As well as members of the Zakłady Kórnickie Foundation, Professor Grzegorz Skrzypczak and Dr. Zbigniew Kalisz. I would like to kindly welcome the Mayor of the Town and Municipality of Kórnik, Przemysław Pacholski, as well as the President of the Scientific Council of the Institute of Dendrology of the Polish Academy of Sciences and the member of the Wielkopolska branch of the Polish Forest Society, Professor Małgorzata Mańk. I kindly welcome the custodian of the Polish Academy of Sciences Kórnik Library, Mrs. Małgorzata Potocka. And the last but not the least, the director of the Góry Stołowe National Park, Bartłomiej Jakubowski. I also would like to kindly welcome the media patronage representatives. They are with us. As you know, the aim of the conference, Research and Practice in Forest Ecology, is to discuss the results of research in forest ecology, taking into account the transformation of forest ecosystem as in local, regional, and global terms. The second aim is to outline directions and perspectives for new research, including interdisciplinary research, and to establish cooperation between scientific centers and practitioners. Our conference is aimed at young scientists, PhD students, and students working in the broad field of forest ecology. We are delighted that so many young scientists from various national and international scientific centers have expressed their willingness to participate in the conference. Our conference participants are from Algeria, Austria, Belgium, Czech Republic, Estonia, Greece, Iran, Italy, Lithuania, Poland, Slovenia, Sweden, and United Kingdom. We hope that the conference will enable researchers to broaden their knowledge with up-to-date scientific and practical information on tree ecology and forest functioning in the light of currently observed and predicted environmental changes. And now please let me introduce our institute in a few words. So last year, just a second. Okay, last year in 2023, it was a very exceptional year for us, for the Institute of Dendrology of the Polish Academy of Sciences, because 90 years ago, in 1933, our institution was established. Thanks to Władysław Zamoyski, the Count Władysław Zamoyski, idea came true 90 years ago. Okay, be calm, please. Tylko możemy mieć podpis. Proszę? Tak, to jest to. Okej. Tak, gra? Dziękuję bardzo. Władysław Zamoyski was the last owner of the Kórnik estate. Zamoyski dreamed of establishing a scientific institution in Kórnik that would conduct research on trees and forests. Of course, the history of our institution, of our institute, goes back much further. 19th century owners of the Kórnik Castle Kórnik estate showed a great interest in dendrology. And it was thanks to them that a very rich collection of woody plant species was gathered in the Kórnik garden, not far from the Kórnik Castle. Titus initiated the creation of woody plant species collection, and Jan, greatly his son, Jan greatly extended the work of his father. Jan Działyński amassed a collection in Kórnik that included around 1,500 species and varieties of trees and shrubs. The last owner of the Kórnik estate, Count Władysław Zamoyski, nephew of Jan, successfully implemented and developed the intentions of his uncle and grandfather. According to the last will of Władysław Zamoyski, in 1925, the Zakłady Kórnickie Foundation was established and the Zamoyski's estate was handed over the Polish state, Polish nation. Zamoyski's will was to establish and maintain a research center both on the mountain slopes and on the plains for the study of everything that falls within the scope of breeding, living, protection, and proper exploitation of all kinds of trees, both domestic in the country and foreign ones, that may be usable for the country. Forest, garden, usable fruit and ornamental trees, their wood, fruits, leaf, and juices. Care to the Kórnik gardens. After some perturbations, on July 1st, 1933, a scientific institution was established in Kórnik, the Center for Research of Trees and Forests, although the establishment of only one of the 4 plant divisions was successful. This is marked in red. The first director of the Center for Research of Trees and Forests was Antoni Wróblewski, who had managed the Kórnik gardens of the Zakłady Kórnickie Foundation since 1926. Antoni Wróblewski was outstanding dendrologist and researcher. After Second World War, the management of the scientific institution in Kórnik was taken over by Professor Stefan Białobok. There is no doubt that Professor Stefan Białobok was the creator of the Institute of Dendrology of the Polish Academy of Sciences. Professor Białobok headed our institution from 1945 until 1979. Rapid development of the institute took place in 1952, when our institution was incorporated to the Polish Academy of Sciences, newly established Polish Academy of Sciences. In the 1950s, the main issues that employees dealt with were pomological experiments, principles of cultivation, reproduction, and selection of poplars, natural variability, systematics and distribution of poplar species, genetics of forest trees, introduction and acclimatization of alien tree and shrub species, as well as systematics and horology of woody plants growing in the wild. Then the first experiments were established in the Zwierzyniec experimental forest, not far from the place where we are right now. It is worth mentioning that the Institute was the precursor of genetic research on forest trees in Poland. In the 1960s, the Institute carried out research covering 5 main thematic areas: physiology of growth and development, seed physiology, genetics, introduction and acclimatization, and systematics of trees and shrubs. Of particular importance during this period was the research initiated by Professor Kazimierz Browicz, covering research of the woody flora of southwestern Asia and the eastern Mediterranean. Kórnik became the center of systematic and chorological research on woody plants, and the results of numerous scientific expeditions were not only publications, but also a significant expansion of the herbarium collections gathered at the Institute. In Kórnik, as the first scientific institution in our country, extensive research was undertaken in the field of forest tree genetics, covering the main forest-forming species, and the results of these studies were used in forestry practice. We owe the initiation of seed biology research and its subsequent development at many different levels to Professor Bolesław Suszka. He worked on understanding the mechanisms determining the dormancy and germination of seeds of many species of trees and shrubs, not only those important for forest management. Research in the field of the physiology of trees and shrubs was significantly deepened in the 1960s by Professor Mirosław Tomaszewski. He developed a research group dealing with the broadly understood physiology and biochemistry of plants, mainly of woody plants, of course. And he initiated research on the mechanism regulating the formation of mycorrhizal associations of trees. In 1974, in recognition of the level of research we conduct at our institution, our institution was elevated to the rank of the Institute of Dendrology of the Polish Academy of Sciences, and we use this name to this day. In the mid-1970s, there were 4 research departments at the Institute: Department of Systematics and Geography, of Introduction and Acclimatization, of Genetics, and of Tree and Shrub Physiology. In the 1970s, the research areas were really very broad. A very important topic discussed by the Institute's employees was the impact of the degradation of the natural environment, especially as a result of industrial activity, on woody plants and the ecological systems they co-create. In the 1970s, it was an era of extraordinary publishing success. In 1970, a series of books began to be published under the common title Our Forest Trees. At the beginning of 1980, the management of the Institute was entrusted to Professor Władysław Bugała, a really famous dendrologist. Employees conducted research covering the following issues: industrial pollution, taxonomic and horological research, seed biology, phytopathology, forestry mycorrhizas, and physiology. In 1986, a new building of the Institute was put into use, which, in addition to the laboratories, housed library collections and an herbarium. In the mid-1990s, the duties of the Institute's director were entrusted to Professor Tadeusz Przybylski. The scientific research program, which is to some extent reflected in the very extensive internal structure of the Institute, was very broad at this time. We had Department of Systematics and Geography, Department of Introduction and Acclimatization, of Tree Genetics, Tree Physiology, Tree Resistance, and Seed Biology. A very dynamic development of the Institute has been visible since the turn of the 20th and 21st centuries. This development coincided with the periods of management of the Institute by Professor Tadeusz Przybylski, Professor Gabriela Lorenz-Plucińska, and Professor Jacek Oleksyn. We conduct research in teams that include 5 scientific departments. They are biogeography and systematics, developmental biology, ecology, genetics and environmental interactions, and symbiotic associations. A great amount of support is given by Arboretum and Experimental Forest, Library, Laboratory of Mineral Analysis, and Herbarium. The Institute employees have always taken care to disseminate the results of their research. They have published numerous books and scientific articles in domestic and foreign journals, including the best ones indexed by Web of Science core collection. This chart, I think, perfectly reflects the publication success of our employees in the past. Our research articles are also noticed by the scientific community. The number of citations to publications created in Kórnik in our institute is growing at a very high rate. The analysis of our achievements also shows how intensively we cooperate with scientists from Poland and abroad, solving scientific problems together. Most of our scientific partners, apart from Poland, represent the United States of America, Spain, Germany, Australia, Italy, China, France, England, and Czech Republic. Right now we conduct statutory research from two disciplines, forestry, and the issue is the complexity of forest ecosystem functions as a basis for their conservation and management in natural and transformed systems, and the second discipline is biological sciences, and the topic is biological basis of woody plants functioning under changing environmental conditions. Our research is funded from a variety of sources. I mean national science centers, General Directorate of State Forests, Polish National Agency for Academic Exchange, Polish Academy of Sciences via Pacific Program, and Institute of Dendrology Research Fund. So this is the amount of money we spend for our research right now in Polish złotych. The collection gathered in the Arboretum were and are extremely important for us as an object of our research. In 2 years, in 2026, we will celebrate its 200th anniversary. The Arboretum is the Institute's flagship. The Arboretum's collection of woody plants is one of the largest and oldest in Europe and is constantly being expanded by the Institute. It is, however, not only a place of research, but also a thriving center for tourists and knowledge promoting activities. The Institute also has an experimental forest with an area about 220 hectares, where many research sites have been established in the past, including numerous provenance experiments, the scientific value of which increases every year. An extremely valuable resource of the Institute is the herbarium, which contains over 56,000 sheets with specimens of woody plants. Many of these resources have been digitized in recent years and are widely available from home computers. The library provides important support for us also. Our collections include over 50,000 books and journals, and many of them are also accessible from your home computer right now. The Institute has always placed emphasis on publishing research results. We also publish numerous journals of our own from the beginning of our existence. We currently publish the Dendrobiology journal, indexed by Web of Science Clarivate, with open, free access to publications. The education of scientific staff was and is the greatest priority for the Institute. We are currently educating young scientists at the Poznań Doctoral School of the Institute of the Polish Academy of Sciences, which we co-founded. We have the right to confer doctorates and habilitations, as well as to notify scientific degrees in two disciplines, forestry and biological sciences. We attach great importance to disseminating knowledge, not only in the form of publications. We have been organizing conferences for years, which enjoy constant interest, as you can see on this picture. We also published numerous popular science articles in biological, in forestry, and environmental protection magazines. And Keeping with the times, we record numerous films and lectures prepared by our employees to shape awareness of knowledge-based society. If you would like to see the movies from the Institute, prepared by our scientific staff, just visit the YouTube of the Institute and familiarize with the presentations. We also prepared some movies about biodiversity, I mean about origin, forest, meadows and city, and the second tour was about evolution, lithosphere, hydrosphere, atmosphere and biosphere. Our achievements have been repeatedly and systematically assessed. We can proudly say that the latest thorough analysis of our institute's achievements places us among the best scientific institutions in the country in both disciplines. Recently, this year, we have been recognized by the European Commission and awarded the HR Excellence in Research logo. The HR logo is awarded to the institutions that provide the best working conditions for researchers and conduct the recruitment processes transparently and in accordance with the guidelines of the European Charter for Researchers and the Code of Conduct for the recruitment of researchers. Much more information and detailed information about the Institute, our activity, about our history can be found in the book of abstracts. I hope you have received, all of you, the books of abstracts, so if you would like to read the deepened story, just read. Okay, so this is all from my side about the Institute. I hope you will be able to visit our Institute tomorrow, on Friday during the workshops. Tomorrow we have lectures, so hard work. So on Friday you will be able to visit our institute during the workshops. You will visit the laboratories and discuss with our staff, research staff at the institute. It would be possible to check what kind of equipment we have in the institute, and if you would like to cooperate with anyone from the institute, we are very open and very friendly for cooperation. So this is all from my side, and now I would like to ask Mrs. Małgorzata Potocka, and Mrs. Potocka will present a short history about Władysław Zamoyski, because this year is a year of Władysław Zamoyski, the Count. The floor is yours.
Good morning. It's a big honor and pleasure to be here and to tell you the story of life, a very short story of life, of Władysław Zamoyski. As you know, this year is the year of Władysław Zamoyski because he died 100 years ago. So in this year we try to tell what he did for Kórnik, for Zakopane, and of course for Poland. But let's start from the beginning. 15 minutes, it's not enough to say the whole history of his life. Władysław Zamoyski was born in Paris in 1853. He was born on 18th of November, and it was a very difficult situation for Polish people because, as you know, in 19th century there was no Poland in Europe. We were occupied, there was a lot of uprising, and it was a very difficult situation. And he was born in a great family because his mother was from the family Działyński. Jadwiga Działyńska. She was the daughter of Tytus Działyński, that man who was the owner of the Kórnik castle and of course created one of the most precious collection of old prints and manuscripts, and the Kórnik castle is one of the oldest library in Poland. His father was Władysław Zamoyski, and it is also very interesting history because Władysław Zamoyski, the officer, the general, the Polish army, was not only the father of Władysław, the son, but he was also the uncle of Władysław Zamoyski, because Jadwiga was, let's say that he was uncle of Jadwiga. So Władysław Zamoyski was brother of Jadwiga's mother, Celestyna. And he was only 27 years older than Jadwiga. So it was very interesting also history, because it was very important in the past to celebrate the patriotism of Polish, and I think that was the reason why they decided to be together, because it was very very important. Here I would like to introduce the grandparents of Władysław. Celina, or Celestyna, or her real name was Gryzelda Zamoyska, and Tytus Działyński. When Władysław was a little boy, he was a baby. He was 2 months. He traveled from Paris to Poznań because his parents decided to go to Turkey to fight, to be very active in politician in Turkey. And Władysław spent 2 years here in the district of Great Poland. So it was his first trip, and we can say that it was a very important trip for him, because his whole life was a very interesting and beautiful travel. Here you can see Władysław as a little sweet boy. He is of course in the center of that presentation, and on the left you can see Władysław as a little boy with his younger brother Witold, and Władysław with his sister Maria. The family Zamoyski, they had 4 children. The first son was of course Władysław, the second was Witold. They had a daughter named Marysia, but she died when she was a year, and their second daughter was also Maria. And Maria was, let's say that the last member of the family Zamoyski who lived in the Kórnik castle till 1937. Okay, so Władysław was born in Paris, and he spent there the beginning of his life. He was very well educated, but as a little child he was of course normal child, so it was a lot of fun, it was a lot of interesting things to do, but unfortunately, as everybody, he had to start his education, of course at the primary school, and his mother also wanted him to learn not only the French history, but also Polish history, Polish geography, literature, and of course Polish language. So we can say that he was very well educated. He could speak French, Polish, and English, but his career as a scientist, as a person who could, I don't know, be somebody important in the educational system, wasn't successful, because when he finished his high school, he tried to start education at the Paris University, Paris Polytechnic, but unfortunately he didn't pass the exams. He tried 3 times and it wasn't successful. At the 4th, he finally passed, but he was the last on the list and he never started his education. From this reason, he decided to go to Australia. So we can say that he left Europe. He was very sad, he was very angry, he was almost desperate. He was 26 when he decided to go to Australia, but it was a great journey. It was a great opportunity to represent the French government and also to see the world. And we can say that it was the beginning of his new life, because in Australia he was a very important person. He was that Władysław Zamoyski from this nobleman family, from this family of Zamoyski and Działyński. And we can say that Władysław traveled around the world. because the first destination was, of course, Australia, Melbourne and Sydney, and villages, little cities, town near Melbourne. And of course, he traveled by a ship. And you can see the original picture of ship Leren, on which Władysław traveled to Australia. He decided also to visit a small island near the Australia. And when he was there, he was a kind of person who tried to show Europe, France, but especially Poland, as a very important place in the history. And he brought from Australia a lot of interesting objects. Some of them you can see in our castle. If you decided to visit the castle, it's possible to see, for example, the head of alligator or different interesting objects. He also brought here a collections of photos, and we have a lot of interesting photographs. But if you want to see Władysław on these photos, it's impossible because we don't have photos with Władysław. But you can see how people lived in 19th century. We can say that Australia was very well developed in 19th century. For example, they had railway, they had a good communication, and in Kórnik in 1879, when Władysław left to Australia, there was no railway station, so we can say that Australia was a kind of a new country, and Władysław had a kind of idea, it's a great place to learn something more, and maybe in future, when Poland will be independent, I can do it in my own country. So it was a kind of a great idea. Here we have collections of different objects, like music instruments, or a bag for water, or beautiful necklace from human teeth, and a mask. It was a mask of cult of different celebration, and beautiful collections of shells. And he sent those things in 5 big boxes from Australia to Europe. But when he was in Australia, he had a kind of idea. Let's go to America, because it's not good to back from Australia to Europe by the same way. Yes, so let's see something more. And he went to Hawaii and of course to USA. And he was also he was very welcome in USA, the same as in Australia, because as you know, people who took part in our national uprising, they need to escape, and they escaped to Australia and to USA. And Władysław was a kind of a messenger, was a kind of a person who was a connection with the history of our country. When he was in Australia, he got the information that his uncle is dying. And the uncle of Władysław was Jan Działyński, the last from the family Działyński, the owner of our castle. And in his last will, Jan gave castle the landscape, the collections of valuable old prints and manuscripts to Władysław Zamoyski. And Władysław back to Kórnik as the owner, young owner, but an owner with great ideas how to change everything. But unfortunately, Władysław was born in Paris, not here, not in Kórnik, not in Poznań. So that was not good for him, because as a man from the other country, he needed to leave the Kórnik, because it was a kind of a Prussian document that people who was born abroad, and Paris, France, was definitely abroad, need to leave our region. That was the reason why Władysław had to move, and he didn't know where. But one day, very popular town in Poland, Zakopane, he could buy this place, and he did it. It was very expensive shopping, we can say, but it was a very good idea because he could stay in that place. His mother moved her school for a young girl to Zakopane, and they spent most of their lives in that place. And in Zakopane, Władysław tried to support the people. He also tried to explain different things. He was a patriot. He also was a very good person. For example, he took care of people. He took care of a culture, and of course, he created, or he decided to build a railway in Zakopane, so a railway station, and he was also a founder of different societies in Zakopane, like Tatrzański Park Narodowy, Tatra Mountain Garden, or different institution. So he was a good owner of that place, and he spent there a lot of time because he could back to Kórnik in 1920. So his trip to Zakopane was a very long trip, and there is a beautiful place, beautiful lake. The name of that lake is Morskie Oko, and somebody in 19th century wanted to take that place, and Władysław was the man who decided to invest his own time and money to protect that place. And because of that man, we can say that Morskie Oko and Rysy belong to our country. So he was also very involved in different objects. And he never married. He hadn't children. He lived with his mother and sister, but there's a lot of legend that his mother was very, how to say it, that his mother told him not to marry, but it's not true. His mother had a kind of idea to find a good wife, but if he had a wife, what happened with the castle and his estate? Probably we didn't have that beautiful, we don't have that beautiful collections because it could be destroyed, or I don't know, or somebody could sell it. But Władysław had a kind of idea, but it wasn't his idea. His grandfather, Tytus Działyński, when he created the Kórnik Library, he said that it is the library for everybody. It is the library for Polish people. And he wanted to give it to everybody. But he died. And Władysław in 20th century decided to continue that sentence. And he decided to give everything to the Polish nation. And from this reason, he established the foundation Zakłady Kórnickie. And it was, I think, that the most important and one of the best ideas of Władysław, because he knew that if he didn't do something like this, the family will took everything and everything could be destroyed. And it was a great and very important for the Kórnik Castle. There is also a lot of interesting history of his life. For example, that he was very rich man, we can say, yes, because he had a castle, he was the owner of Zakopane. But for example, you can hear different interesting history that he used to sleep on a desk, not in a bed, because he couldn't accept after the 1918. He couldn't accept the stupid of Polish people. And he wanted to sacrifice his own comfort of sleeping and decided to sleep on a bed, on a desk. So we also know the history of Władysław, who traveled in a train in the third class because he wanted to live very, I don't know, he preferred ascetic style of life, yes, and he traveled with his own food, but nobody wanted to eat those things because they didn't taste good. So we have a lot of funny history of his life, but we know that he was patriot, he was great man, and because of him we can still visit the castle with beautiful collections of furniture, of different objects. We can still see those collections of old prints, manuscripts. We can visit the garden, arboretum, and we can say that the Kórnik, because of Władysław and his family, is the center of culture, history, and it's a very good example and lesson for us how to be patriot. So I know that it's maybe not enough because Władysław died in 1924, so we can say that his life was very long. But if we want to say something more, we need to say a lot about travel to Australia, about travel to England, or about his childhood when he was in Turkey and he was a great soldier as a little boy. But if you want to know something more about Władysław, I would like to invite you to our castle and you can hear more interesting history of his life, because the castle is his place and he's still there. Thank you.
Thank you very much. We have concluded the official part of the conference. Now we are moving to the lecture segment. I invite Professor Marcin Dyderski. Professor Dyderski will discuss future of European tree species, how will climate change shape forest.
All right, thank you very much for invitation. Let's look into the future of forests in Europe. We have a lot of studies about species distribution models, and it became something very common. In 2012, Dave Richardson, in editorial to Diversity and Distributions, wrote that every person and their dog have now access to data, to Maxent models, and are able to do species distribution models. It's true, I can sit near the computer with my dog, and within 1 hour I can develop a model of distribution to every species on the Earth, because now we have data. But if there is so easy to do a model, just a model, is it applicable? Has it sense or not? Let's dive into the process of model development and how to do it properly, because of the thousands of publications that I've found today morning, that we have over 9,000 of publications using just only one phrase, Maxent, method of the models distributions. But not all of them are proper, are valid, and how to make a good model. We will use this scheme to show what is necessary to build a model that will help us understand how to predict the climatic optimum of the species, and how to make a prediction using the future climate conditions, how it will change. On the left side, we have two parts of the input. We have our predictors, climatic variables, and we have our dependent variable, data about species distribution. We can use different variables describing climate, but what is important? If we want to look into the future, we need to take these variables that we have future values, the predictions for the futures. Also, most of variables describing climate are correlated to each other. And if we put all of them into the model, some of them will be explaining each other and will lead to the inflation of our model and will not allow us for valid conclusions. What to do? We can make just a simple correlation matrix and exclude highly collinear variables with, let's say, the Pearson's R coefficient about 0.7 or higher. What are these bioclimatic variables? This is the set of most commonly used variables. The data about monthly temperatures and precipitations are used to calculate all of these variables, and this is from meteorological stations interpolated for each pixel of the world. We have this gridded climatic data, easy and available in the databases like WorldClim or Chelsea, but we need to remember that they can be collinear. Also, for these bioclimatic variables, we have future predictions. But what is the future? The future is unknown. We don't know exactly how the future will look in different time horizons. And taking future values, we cannot just randomly or subjectively select one scenario. We need to include three main sources of uncertainty. First of all the climatic variables for future are calculated using atmospheric global circulation models. And these global circulation models are calculated using different algorithms. So we need to take it into account, and we need to use not only one. In the results part of this presentation, I will show you why it matters so much. Another issue is timeline. We can make a prediction for the nearer or further future, and it also makes a difference in the prediction of changes of climatic optimum. And the most important source of uncertainty is climate change intensity. We don't know how much efforts we will put into stopping climate change, stopping emissions. So we need to use a range of scenarios to show the range of possibilities that can be when we talk about the future. In the fifth report of IPCC, you can see that there are different scenarios like RCPs, representative concentration pathways. This is the value of radiation enforcement expressed in watts per square meters, and this report assumes three main scenarios. I call RCP 2.6 optimistic. It's very optimistic because it assumes that in 2100 we will have more or less the same CO2 concentration as we already have, and it will lead to the change of mean annual temperature globally from 0.2 to 1.8 Celsius degrees. There's also moderate and pessimistic scenario called also the worst case, which we can use to see what will be if we will stop climate change, what if we partially stop climate change, and what will be in the worst case if we will do nothing. These scenarios are widely used in the publications, but 3 years ago it was published the 6th assessment report of IPCC that used not RCPs but SSPs, shared socioeconomic pathways. These scenarios are accounting for not only emissions but also population growth, land use changes, and many other socioeconomic and political factors. And let's look on SSP 1 to 6, which is called sustainability, the most optimistic case. It assumes much higher concentration of carbon dioxide and much higher temperatures. These scenarios have different names. but more or less are the higher is the number of the SSP, the more pessimistic the scenario is. So we know something about the climate data. We also need data about species distribution. In all cases, data about species distribution are unevenly sampled. So we need to make something to reduce the effect of sampling effort. Because if we have 1,000 points per square kilometer in Germany and 100 in Poland, the first region will be overrepresented in the model. The simple solution is to make a regular grid and randomly select only 1 point per grid cell to get refined occurrence data. With distribution data, we have to remember that in the perfect case, we should have presence-absence data. It's really hard to imagine to get presence-absence data about tree species distribution in whole Europe. Like we had to go everywhere and say, okay, here there are no Scots pine. So the real case that we are using is presence and pseudo-absence. Outside the distribution, we are randomly selecting some pseudo-absence points. In the case that Richardson wrote about every person and their dog, the primary source of data is Global Biodiversity Information Facility, GBIF. But GBIF is not ideal source of data. There are some errors, and also some regions are underrepresented, especially in Global South. But even in Europe, you can see a difference in data coverage. So what do we usually do? We are supplementing data from GBIF by many other sources, especially citizen science, which can be very helpful in filling gaps. Also floristic inventories, some basic papers, and gray literature can be a good source. Regardless of what data, what species we have, if you want to get a valid conclusion, not the best conclusion, but the valid, we need to cover at least one edge of species range to get a gradient of species presence, absence. Here is an example of some usual case of data. In the left side, you can see English walnut distribution mapped in 2018, and the red line showing areas with higher and lower sampling intensity, and some examples of sources that are not in the databases that are easily available, but you need to manually georeference these maps and include these data points to increase the model's quality. Okay, model. What is the model? There are different options. I'm talking about maxent because maxent is algorithm that in its assumptions is dedicated to handle pseudo-absence data. Other techniques like GLMs, GAMs, machine learning techniques like random forests or GBMs are assuming presence and true absence. Also, many papers are using these other techniques to use pseudo-absence data, but not always it's valid. Regardless of the technique we use, the very important thing is to not develop a model on the whole data set we have, but to split it. Use, let's say, 20% out of the training data, out of the data that we use to model development, to independently evaluate the model, to check whether model is overfitted, whether we can extend the conclusions from the model to other areas. And also, when we predict the future distribution, in some papers, it is just running a model, which means developing a new model using the same response variable, but with the future predictors, And this is highly biased approach. The correct way is to calculate model responses from the model built on current conditions and use the predict function to get a prediction, not to build a new model. Why we need model? We have a continuous response, predicted climatic suitability. It is not a probability of species occurrence because we are using only climatic predictors. We are not accounting for many other factors. And this climatic suitability is continuous. And we need to say where is the threshold of presence absence. And when we calculate the threshold from the model, and we will compare the predicted suitability in future and in current climate, we will obtain a confusion matrix showing the possibilities of current and predicted model response for each pixel. And based on this discrimination, we can say where we can expect range contraction, the decrease in habitat suitability, where we can expect range expansion, sites that are currently not suitable to occur for the species and in future could be colonized, where we can expect persistence of the species. It looks nice, but we also need to look not only on the model results, but also we need to look inside the model. Because if the model will put out the map, okay, we have a nice map, but what is behind it? It's a black box. And we don't want a black box, because if we have black box, we don't know whether model is doing something that has ecological sense. So we need to look into variables importance to see which climatic variable is most important for the model, and for partial response curves to see how the predicted climatic suitability changes with the change of predictor values. It shows us, like for Bio 18 on the bottom row right side, that with an increase of this variable, the climatic suitability increases. And this is the sum of precipitation in the driest quarter, and it shows us what is the limit value, and that this variable is limiting the occurrence of particular species. So with these tools, we can not only say, okay, we have model, it will be in the future, but we can say why we think that it will be in the future, and whether the model response is related to the biology and ecology of species. We can explain it with reference to the studies about physiology, about seed physiology, and we can give the model a good ecological background. Okay, enough talking about models. Let's go to some results. I will briefly show what are the outcomes of our studies on trees distributions in Europe. First study assessed responses of 12 forest-forming tree species in Europe in 2018. And we predicted that conifers will mostly lose most of its current distribution, even in the RCP26. RCP26 is the optimistic one. So looking on the Scots pine and Norway spruce, we can say that it will lose most of its climatic suitability, even in the optimistic case. Optimistic is not realistic, because we already passed the threshold of 410 ppms. So we are rather between RCP 4.5 and 8.5. But good news is that broadleaf tree species would persist mostly. The exception is Quercus robur, and the difference between these two oak species is very significant here. If you are more interested in this result, Epsilon company provided within a Data for Good program an application that you can use this QR code or link, go into the application and zoom our results into the region that you are interested the most. All right, the next step that we took was assessment why global circulation models are so important. We used English walnut as a model species, and we checked differences in response for 3 different global circulation models. There are rows in this matrix of charts for different climate change scenarios. And if you look on them, the differences in the predicted suitable area for walnut are very different across these 3 GCMs. We can see that HAD GEM M2ES is more pessimistic, MPEI is more optimistic. So if you do species distribution model and you use only one GCM, it will be biased. The good option is to make prediction for each GCM separately and then average them. We also wondered what about shorter time horizons. To do that, we go back to the results for Robinia pseudoacacia, and we did models for 2050s and 2070s. And you can see that the most of changes, especially the range contraction showed on orange, that we predict for 2070s, is likely to happen in 2050s. It means that we have 20 years less than we previously predicted to face the changes. Some people think that the solution to climate change can be using alien tree species. We did also models for the most common alien tree species in Poland, and you can see that alien broadleaf tree species on the left side will mostly persist and will expand their potential ranges, except Quercus rubra, but alien conifers, especially Douglas fir, that is expected to be the tree of the future, will lose their climatic suitability. The exception here is Pinus strobus. But if we have models, models are only models, and we need to understand what is in the model to be conscious of the limitations. And models are showing us only wide-scale responses to climate. Wide scale, it means that we have pixels 5 by 5 kilometers, and model is not accounting for the microclimatic, microhabitat diversity within that. And not includes biotic agents. I showed here Fraxinus excelsior as winner, but we all are conscious that ash dieback is decimating the ash stands across Europe. Also, it does not include dispersal limitation and is prone to data-driven limitations, so we need to be conscious of all limitations. What will be the consequences of changes in trees distributions? It will change dominant fundational tree species in forests, which will affect nutrient cycling, light transmittance, affecting then all dependent organisms like fungi, soil animals, epiphytes, leading to shifts in plant communities, biodiversity decline, and loss of ecosystem services. And from the forest management point of view, we expect the retreat of high productive tree species, especially conifers, that are crucial for forest management. It will result in lower wood supply and lower wood quality, bigger needs for reforestation regeneration, and maybe it's not so important, also will affect non-woody forest products. So, instead of classic conclusions, I would like to stress out that everyone can now develop species distribution models, but it's not just, okay, I have model, it's fine. We need to be conscious what we have in the model and what are the limitations. Because, as Cox said, all models are wrong, but they can be useful when they meet the logical assumptions, are ecologically interpretable, and when we remember about limitations. We predict that climate change will affect tree species distributions at an unprecedented rate, and it's already started to happen, especially when you look for the Scots pine in our region. Even widespread species like pine or spruce can be threatened, and future forests will be shorter and composed of mainly broadleaf trees. And having this in the mind is crucial to properly plan forest management and nature conservation. And thank you for your attention.
Thank you very much, Professor Tyderski. I invite Professor Andrzej Jagodziński. Professor Jagodziński will discuss carbon storage, examination, and forest ecosystem research and practice. Please.
Aha, dobra. Okej, dobra. Dzięki. Dobra. Okay, it works. Okay, so I would like to tell a short story about carbon storage estimation in forest ecosystems. This is a very important issue because we are talking about climate change, about global climate change, and those data shown by Professor Dendrology are very linked with the data I will show you in a moment with the carbon storage, carbon pool, carbon estimation. The importance of forests cannot be underestimated, and this is a global truth. As one of the most important components of the terrestrial ecosystems, forest occupies right now about 31% of the globe, of the land surface. They provide many ecosystem services, as was mentioned in the previous presentation, which are essential for humans and for our life and for survival of people. Forests are critical habitat for biodiversity, and they play an essential role in its protection as host of many different species. I mean about plants, animals, about microorganisms, fungi, etc. They serve as vital shields against extreme weather conditions, regulate water supplies, and they prevent erosion by reducing the force of rainfall and by absorbing water. And what is really important, they influence regional climate. They can make it. And forests are also a resource of timber and non-timber forest products, and many more functions belongs to the forest ecosystems, describing the importance of forest ecosystems. Forests can help to stabilize the climate and play a really crucial role in mitigating climate change, but acting as a carbon sink, absorbing atmospheric carbon dioxide. But this is not the only one way how it works. They account for more than about 60% of total biomass of the globe and over 50% of carbon stocks on a global scale, I mean, in living biomass. And they are a major contributor of carbon uptake and storage. Forests absorb a lot of carbon dioxide from the atmosphere during the photosynthesis process and return a large part of the fixed carbon back to the atmosphere through respiration. However, a fraction of this carbon, assimilated carbon, is stored in above and below ground biomass, litter and soil. And the question is, do we have any precise tools to estimate the carbon storage in forest ecosystems? Usually we estimate in the most easiest way the carbon storage in tree stands. It's more difficult to estimate the carbon storage in forest soils, in litter or herb biomass. We have a lot of tools for determination of carbon storage in trees. So I will focus on trees during this presentation. Many years ago, 10 years ago, I have read a very interesting paper by Keenan et al. It was published in Forest Ecology and Management. Based on the data presented by the authors of this paper, the forest area diminished on the global scale during the 25 years period of time studied by the authors by about 3%, whereas in Europe and Poland, forest area increased. And 10 years have passed away, and the Earth forest area continues to shrink, particularly in Africa and South America. And we can see on this picture, this new one from 2021, that the area of forests in Africa, South America, North and Central America is diminishing. On the other hand, we know that the atmospheric carbon concentration is still increasing from the beginning of the industrial area. Now it is about at least 420 parts per million in the atmosphere, and this amount is still increasing. So the question is how we can reduce carbon concentration in the atmosphere. And we have some answers, some solutions. We can reduce carbon dioxide emissions from human activity. This is the first possible activity, or increase carbon dioxide capture and its accumulation, for example, in forest ecosystems, or both of them. But the first action is undoubtedly crucial for the life on the Earth. In the Web of Science database, we can find many papers about carbon storage in forest ecosystems, carbon sequestration, not only in forest ecosystems, of course, but on many terrestrial ecosystems, the methods how we can determine the carbon storage in different communities. But there is a lot of questions if we are precise as the most possible, if we precisely determine the carbon storage in forest ecosystems. In the Web of Science database, we can find many papers about this issue in forests, and the number of studies is still increasing. A lot of papers are published every single year. But most of these studies are related with two components of forest ecosystems. I mean about biomass of trees and about the soil. But the number of papers about carbon storage in soils is increasing more than on carbon in the biomass of trees. In Poland, we have many results of previous studies about carbon accumulation, carbon storage, carbon pool, carbon distribution in forest ecosystems. So we may use our own data for estimation carbon storage almost in every single forest stand. But the question is how precise we are using those data. And now I would like to present some results from the research of our team from the Institute of Dendrology with many other partners from other institutions. And the project title was Remote Sensing-Based Assessment of Woody Biomass and Carbon Storage in Forests. As I have mentioned before, there are many ecological processes that create carbon flux in forest ecosystems. I mean about assimilation, respiration, accumulation, decomposition, and soil respiration. However, in a global or country perspective, not the single tree or a single stand, we should focus on more general data to determine carbon storage in forest ecosystems and its changes over time in relation to the forest management practices. So the question is why we should focus on biomass. First of all, the biomass is more variable than carbon content. It is a derivative of tree stand dimensions, and it is easy to estimate based on forest inventories. And almost every single country in Europe has its own inventory right now, so we may use a huge databases to estimate the carbon storage. But the question is about the tools we will use for determination of carbon storage. In our study, we have collected data on above-ground biomass for almost 3,500 of sample trees on more than 4,000 stands up to 140 years old. Research sites, as you can see from this picture, cover whole Poland and thus the different habitat conditions. We studied the 8 tree species, the most common tree species, forest-forming tree species in Poland and also in Europe. I mean Scots pine, Norway spruce, white fir, European larch, pedunculate oak, European beech, silver fir, and black alder. So let me describe briefly the study. Of course, everything starts in the forest and ends in the forest. We measured diameters and heights of trees within each tree stand. Based on diameter and breeze height distributions, we have selected 8 model trees in each stand, and the sample trees were harvested, divided into components, I mean about needles, cones, fruits, branches, stems, and weighed in the field to obtain the fresh biomass of every single component of a model tree. In the laboratory, all the samples were oven-dried, and then dry biomass was measured. So we calculated the amount of water in every single sample. So as a result, we have determined oven-dried biomass for all the sample trees harvested from those more than 400 stands. In each biomass sample, we have determined carbon content in the institute, and then based on this data, we have developed site-specific allometric equations for biomass determination for each stand, so for every single stand, and we have calculated stand volume, I mean about merge table volume, stand biomass, and as a result, biomass conversion and expansion factors. I will describe this trait later. The biomass conversion and expansion factors for each biomass component and total above-ground biomass of every single stand. So, as you can see from these pictures, a lot of work in the forest, in the field, and a lot of work in the laboratory. So it took a lot of years. I have started my trip with carbon estimation in 2002, so really many years ago. Based on the sample trees, the model trees collected and the features and its dry biomass, we developed for every single stand so-called site-specific allometric equations for biomass determination, which are specific for a particular study plot. As you can see on the picture on this graph on the right down corner, there is a close relationship between the diameter at breeze height at 1.3 meters above the ground and above ground biomass. This is the easiest allometric equations we can see. So based on this mathematical relationship, we can estimate the Y value based on dBH. Sometimes it's better to use other tree features like height of the trees, like the length of the crown, many other features, but for many cases, diameters at breeze height will be enough to estimate precisely the biomass and carbon accumulated in the biomass of a single tree. Based on a sample tree's biometric features and its dry biomass, we developed this kind of site-specific allometric equations for determination of biomass for every single plot, and then using the tree stand measurements, I mean about the diameters at breeze height and heights if needed for every single stand, we calculated dry biomass of each tree and we summarize its per unit area. As a result of this part of our studies, we calculated regional biomass conversion and expansion factors to estimate above-ground biomass and carbon storage. As you can see from this picture, we have to divide the biomass of the tree by the volume or merchantable volume of a tree. So the unit is tons or megagrams per cubic meter. And now I would like to show you some results from our studies to show some uncertainties and some thoughts we have about the model we have developed based on these studies. Just for example, we have compared our site-specific allometric equations for biomass estimation data for large, about European large, with other published data for the species for different biomass components. As you can see from this picture, we have divided the tree, every single tree in the forest, into stem bark, stem wood, branches, foliage, stem, above-ground woody biomass, and total above-ground biomass. We have also data in our institute collected for many years related with below-ground biomass, but this is really time-consuming work. So in this research project, we based only on the above-ground biomass. So we divided the tree into components and calculated every single component and expressed in oven-dried biomass. As you can see from this figure, our data for above-ground biomass, this is in the right down corner, are higher than from other studies published earlier and funded by us in the literature. So using allometric equations developed in other site conditions or maybe in other management practices may lead also to biomass underestimation and sometimes overestimations. It might be different. And similar results we have obtained for silver fir, Abies alba, and our data for above-ground biomass also are higher than from other studies. Let's move to Scots pine. This is the most important tree species right now in Poland. Yes, I'm afraid and almost sure after the first lecture that this species will not be so present and so common like today in the nearest future. As I have mentioned, similar results were found also for the most important tree species in Poland and Europe. So using allometric equations developed in other site conditions or other management activities, practices, may lead also to, as I have mentioned for the first and the second species, underestimation or overestimation also. So using allometric equations developed for other regions, and we have a lot of data from Europe, but about 15 years ago we have a lot of data for Scots pine grown in the northern conditions, but almost no allometric equations in the central part of Europe or in the southern part of Europe. But right now we have a lot of this kind of allometric equations and other functions that help us to estimate carbon storage in forest ecosystems, mainly in forest stands. Using allometric equations developed for other regions may also lead to a really very high errors. So we are not sure if our results are close to the reality. For example, for young Scots pine stands, the error may be even of 50% of the observed biomass, and it was also published in this paper. Okay, so the carbon content in tree biomass is less variable. I have mentioned at the beginning of my presentation that biomass is more variable, and this is why we estimate the biomass, and carbon content in trees is less variable. So here we can see results for our analysis based on over 27,000 of samples of different parts of trees. So as you can see, these data for 8 the most important tree species are different than data prepared and presented by Intergovernmental Panel on Climate Change. According to the data from Intergovernmental Panel on Climate Change, mean carbon content in the above-ground biomass for conifers is 51%, whereas the broadleaf trees, the value is 48%. And we can see that the distribution, that the range is very high. As we can see from this picture, and these IPCC values are too high for most of the conifers and too low for broadleaf tree species in comparison with our data. And now let's check how stand carbon mass change over the stand age. There are a lot of papers about relationship between carbon storage in the biomass of tree stands and their age, and many other features of the stand. I mean about basal area, mean height of the stand, mean diameter at base height of the stand, and many other, and stand density. So we can see a clear trend for all of the species studied, 8 species studied. However, carbon mass for stands of the same species might be different, even if the age is the same. So there are some other features that we should take into account to estimate biomass, probably. However, if we use stand merchantable volume per hectare, we may relatively well determine carbon mass stored in the stand's above ground biomass. As you can see from these pictures, the relationship for every single species studied by us between the carbon mass per unit area and merchantable volume per unit area is very high. The relationship is very high. So our predicted values of carbon storage in the above ground biomass are more precise than data from other research studies when we use them for our stands. So it's better to use our own data, our own original models, to more precisely estimate the carbon storage or biomass in forest ecosystems, this part of forest ecosystems. And now let's compare our biomass conversion and expansion factors with those published by IPCC. As you can see from these pictures, for 3 different groups of stands with different growing stock, our biomass conversion and expansion factors are lower than published by IPCC. Thus, to precisely determine local biomass and carbon storage in forest stands, we should use regional models for biomass and carbon determination for every stance. Thus, we need local solutions to precisely determine carbon storage in forest stand biomass. And now I would like to show you some data for large and fair stands in comparison with other models. As you can see from this picture, we have different models from the literature data, and we have compared the biomass of many different stands, estimated based on our results and estimated based on many different models established in Europe. And as you can see, the differences are really very huge. And above ground biomass of the stand studied estimated using values provided by IPCC guidelines are distinctly higher than determined by our local models. And the variability of biomass conversion and expansion factors is geographically based and related with climate conditions. So if climate conditions will change, it is possible that not only the stand composition will change, but also many other features of tree stands and forest ecosystems as well. So biomass conversion and expansion factors, those values published by IPCC, are based on a really very small number of papers from entire Europe. So this is not the way how we should precisely estimate carbon storage in tree stands, because those data based on, if I remember well, on 8 papers, on 8 papers, and 1 was prepared in Europe. Thus, using this data is not the best solution for carbon estimation storage, for carbon storage estimation in forest ecosystems on a regional scale. And the variability of, as I have mentioned, the variability is very visible. But now, based on those data we have collected in Poland, we develop regional solution for proper biomass estimation. So a set of biomass conversion and expansion factors for above ground biomass. This is only one of the factors. For example, if you need to estimate above ground biomass of 50 years old beech stand, just multiply the red value by the merchantable volume of the stand studied. So this is very easy in this way. But of course, probably we will have some differences with the reality, as I have mentioned before. And these are biomass conversion and expansion factors for carbon stored in the above ground biomass of tree stands. For example, also the same like in the previous picture. If you need this to estimate above ground biomass of 50 years old beech stand, just multiply the red value by merchantable volume of the stand studied. And comparing biomass conversion and expansion factors stands with volume higher than 200 cubic meters per hectare, we can see that our local biomass factors are lower for all the species studied in comparison with IPCC values. So I will show you in a few words the application, how we can use those data. We use the forest data bank. We have something like that in Poland, so we can check the data for more than 3.5 million of forest compartments. We use the data from those database, and we have estimating carbon in tree stands in Poland. So as you can see from this table, the carbon mass is about 600 million tons in forests, in standing biomass of trees in forest stands. And 50 conclusions. Carbon storage in forest stand depends on the tree stand parameters, in many tree stand parameters, of course. Forest inventories are a good source of data which may be used for country-level carbon storage reporting. However, generalization of models is limited, thus regional approaches provide more reliable results. Biomass conversion and expansion factors are quickly decreasing and stabilize in stands older than 20 years, and this is species-specific and correlated with wood density. Our approach may be applied also to data collected using remote sensing way, and further investigation are important for recognition carbon resources in other components of forest ecosystems, I mean about soils, dead wood, forest layer, etc. And at the end, 3 questions. How possible changes in European forestry species distribution may change carbon storage in forest ecosystems? This is in relation with the previous presentation. What activity in forest management practice may help to increase carbon storage in forest ecosystems? And the 3rd question is what scientific activities should be done in the future to estimate more precisely the significance and potential of forest ecosystems in carbon dioxide mitigation? Thank you so much for listening to the presentation.
Thank you very much, Professor Jagodziński. Invite Professor Daniel Chmura. Professor Chmura will discuss contribution on quantitative genetic to adaptation, forest to climate change.
Okay, thank you so much. Where's my presentation? Okay, good morning, everyone. I work with quantitative genetics, and I'll try to talk about its possible contributions to adapting forests to climate change. So without further, well, let me check this equipment. Okay. So today I'm going to briefly tell you what quantitative genetic is and what it does. And also we'll talk briefly about climate variability and climate change and how those changes may affect trees and forests in terms of threats and opportunities. And I'll tell about methods how we study tree species adaptation to climate change and some options we may use that may help forests to adapt to those changes in climate. So, quantitative genetics is basically the branch of genetics which deals with phenotypes. In quantitative genetics, we very rarely look into genes, but instead we measure traits. We look at the traits most of them with continuous distributions within populations. And we use the networks of prominence experiments or common garden experiments, which are established in specific layout, which lets us to use statistical methods to link phenotypes to genotypes. And we, from that information, we infer information about the gene action behind the observed variation. And information from those experiments may be used to guide management decisions regarding adapting forests to climate change. But before we do that, we first need to understand climate variability and climate change and what it may mean to trees and forests. Of course, probably we all know that climate is naturally variable. The climatic system is naturally variable. In the past, there were periods of both cooler and warmer periods, glacial and interglacials, when climate was warmer or cooler. But the current climate warming is unprecedented in the geological timescale. It's very fast, and we have a lot of evidence that it's related to human activity. However, there is the big deal of uncertainty how the future develops, and we really don't know. I don't know if it's can be seen. And we really don't know how warm the climate will get. We have the idea about the direction of change. We know that climate becomes warmer, but we don't know how much warmer it will get. And there are some associated changes also in frequency of extremes, climatic extremes. So we know that the climatic system, well, we expect that climatic system will be less stable, will be more with more extremes, and perhaps drier at some areas with changed distribution or patterns of precipitation. And trees and forests will have to function in those changed conditions. There are, of course, some opportunities related to tree growth in warmer atmosphere, but they are guarded by some fine print. It's not visible, I guess. So we may observe longer growing seasons or better tree growth, provided enough moisture, enough nutrients are available, and if phenology will not be disturbed. And all these changes in climate overall lead to increased stressors, increased stresses and threats, both abiotic and biotic, which from the human perspective lead to the negative consequences. The trees will be more vulnerable to stresses, and basically they will be weakened, and we may also expect some disturbances in ecosystem interactions. But when we think about the responses of trees to climate change, we need to distinguish between the reaction of individual trees and tree populations, because individuals may only acclimate to changes in the environment. They modify their physiology, they modify their morphology, so that to better perform in their environment. So within their phenotypic plasticity, they may react to some range of changes in the environment. But when we think about populations, we have to consider processes which happen across generations, not only within the current individuals we have on the ground, but also across generations. And of course, populations can move, can migrate if new areas become favorable for growth. Tree populations may expand, and we know that it happened in the past after glacials tree species occupied new areas as climate became more favorable in new areas. And of course, there is adaptive evolutionary adaptation within populations by changing allele frequencies within populations, between generations. Populations become adapted to the prevailing conditions. And if those natural mechanisms don't work, then populations may just go extinct. How do we know what would happen to trees in future climates? We've heard in the first presentation about species distribution models, and there are a lot of, well, a lot. There are some scenarios, different scenarios of or projections, what would happen to tree species and basically to the suitable climatic niche. In species distribution models, we talk or we consider species, species climatic niche. We don't know exactly what would happen to those species. And there are important limitations of species distribution models because they are useful and, well, they are built for the rough spatial scale. And in this big spatial scale, from global to regional spatial scale, the climate is the most important factor affecting species occurrence. But if we go down into finer spatial resolution, then other factors gain importance. And it's okay because, well, as Professor Duderski mentioned, these models just ignore those factors, but those models are built that way, so they are useful for that purpose. But most importantly, they ignore, well, they treat the species as a whole, and they ignore adaptive genetic variation which is within the species. And we know from provenance studies that there is a lot of genetic variation within the species which is related to adaptive ability. The provenance experiments are kind of field experiments where we collect seeds from various stands growing in various conditions, and we bring them together and plant them in uniform conditions, and we repeat that design in several locations. And that allows us to study the causes of observed variation in traits and ascribe this variation to genetic and environmental parts. But most importantly, when we move those populations from different locations to some other places, they experience climate change. And that causes that provenance experiments are good models, not perfect, but good, for studying adaptation to climate change. And this idea, well, the idea of eco-transfer was the idea by Professor Matyas from Hungary. And his eco-distances were based on precipitation, latitude, and heat sum. That's why they were a little bit confusing. But the idea is quite simple. If we describe population origins and planting sites by some climatic factor, we may calculate transfer distance based on that climatic variable as a difference between planting site climate and population origin climate. And there are two models basically to use this transfer distance. Okay, this is the example of probably the largest provenance experiment in the world, with lodgepole pine in western North America, with 2 subspecies. Basically, there were 140 populations tested in this experiment and over 60 planting sites. Of course, not all populations were planted everywhere because it would be impossible to handle such a big of experiment. They measured tree height in all available planting sites, and the first method to study adaptation is to focus on the reaction of individual populations, the population to planting site condition. This is called response function in the middle. This is in the middle graph. So we have the trait of interest here and climatic variable of planting sites. So this is the range of annual temperatures of planting sites where those populations were represented. And each curve represents the reaction of individual populations to that planting site's variability of climate. This might be any climatic parameter. It just happens that it's annual temperature. And the other method is called transfer function. And we focus here on the reaction of multiple populations to the transfer distance. The transfer distance is, as we remember, the difference between planting site and origin site. And this very well show the adaptation, because usually the populations close to 0 transfer distance, meaning that they are local to that climate, usually they are somewhere close to the optimum performance. But not always. As you can see here, for the Contorta subspecies, the optimum performance here is quite off of 0 transfer. So it means that those populations don't usually occupy their optimum climate. And if we integrate these 2 approaches, we can create something called universal response function, which takes into account both planting site climate and provenance climate. So I'll not talk too much about details about that. And we used the transfer function approach to study the response of Scots pine and Norway spruce in a much smaller spatial scale. In Poland, we analyzed available provenance experiments of these species in Poland, and we created those transfer functions based on mean annual temperature and annual precipitation sum, and we didn't find any significantly important pattern for Scots pine, but we found a negative effect on above ground biomass carbon production in Norway spruce when populations were planted in locations which were warmer and drier. And this is the same for European beech. This is the study under review right now. And we found some positive effect on population productivity when planting site was warmer by up to 2.5 degrees Celsius, but the reaction was curvilinear, so this was the limit. And we also found that one site was weighting heavily on those curves, and after exclusion of that site, we didn't find any significant trend for transfers based on annual precipitation, annual heat to moisture index. But there was a negative effect of planting populations in conditions which had drier summer. Okay, and if we have information from those provenance experiments, so basically the relationship between climate and some trade performance, we may use that information to spatially plot the distribution of the trait. So similarly to species distribution models, but instead of projecting species occurrence, we may project the distribution of the trait of interest. Usually these are traits which are affecting fitness, which are important for adaptation. And here you have an example for common beach of spatial distribution of probability of young tree survival. And from the same study, this method may be used to refine the projections of species distribution models, which are based on climatic variables only. So on this bottom graphs, you have here future habitat suitability for European beach projected based on climate only, and here based on probability of young tree survival. And you can compare these two graphs. And for example, like in this area, northeastern Europe, you can hear the high habitat suitability based on climate only, but not on tree survival. But in the Western Europe, the projections are not that bad if you take into consideration probability of tree survival compared to climate only. And information from provenance experiments about the trait variability may be used to guide decisions in forest-assisted migration. Forest-assisted migration is proposed as an option to help forests to adapt to climate change, and usually it's framed as an assisted population expansion or assisted gene flow within current species distribution range, in contrast to assisted range expansion or species migration when species are moved beyond their current geographic range. So assisted population expansion is in fact moving genes or individuals from climate which have now conditions which are projected in the future somewhere else within the species distribution range. And this is the active area of research, mostly in North America. And so this is an example on the right from that study, one of the studies done there. And forest-assisted migration fits into the whole suite of adaptation strategies focused on ecosystem resistance, resilience, or transition. And of course, they are more compatible with strategies related to transition and resilience because they accommodate some degree of change in forest ecosystems. So to conclude, the climate is naturally variable and we are more certain about the direction of change, but it is changing, sorry. And we are more sure about the direction of change than the extent of that change. And quantitative genetics may help forest adaptation to those changes by identifying the extent of variation within the species, extent of adaptive ability within the species, and by identifying the seed sources and planting sites for assisted migration. And I didn't really touch on selection and breeding for resistance, but selection and breeding are kind of traditional focus of attention in quantitative genetics. So that's why I didn't discuss that. And thank you so much.
Thank you very much, Professor Chmura. Now I invite Professor Wachowiak. Professor Wachowiak will discuss genomic and population history and adaptation variation in forest trees.
Sorry, I can't find this. Właśnie próbuję. Ekran jest z lewej. Okay, good morning. It was long morning, and well, I hope you are still here. And good news is that we have 20 minutes to coffee, so it's close. Retrospectively, hearing the previous talks, I had impression that whoever put us together did a very good job, because I think this is very much complementary to what was already said. And my idea is to show you a little bit of genomics perspective on population history and adaptive variation in forest trees, which I think is very much related to really everything what was already said. So I hope that will be a nice kind of contribution to the topic and explanation maybe of some things from genetic perspectives. And I had many thoughts, I would say, hearing the previous talks, that it's really complementary, and I hope maybe after all we'll have some nice discussion how to look at different sorts of data and make advance in our fields. I think that will be some space for that. So anyway, when you think about, sorry, I hope it works. Okay. So whatever species you study, it went through some speciation event, yes? So at the beginning we had some baseline populations, and along the time, which is happening of course in changing environments, this population usually is split to some other smaller populations, depends on the isolation, yes? Some of them will die, some will go through, and some will, let's say, shrink their distribution, some will merge, and some further split, yes? So this really what we observe at the moment, whatever system you study, whatever species, like in this Scots pine, yes, we are here, yes, but the species had its own history of climate change, environmental change, divergence. For Scots pine, we estimate the speciation for this species to be more or less 5 million years ago, yes, so it means it survived, and not only survived, but very much succeeded, yes, so thinking about different models, maybe we should also think about this, that even though there are really dramatic climate changes, the species is still here and it's doing quite well, or maybe not as good as before, but still is here. Yes? So it's important to know that we are here really, but many things happened in the past. So when you think about genetic variation of any species, we can divide this to two components. One will be neutral, so background genetic variation, which is related to population history, like population range changes in the last 20,000 years. Let's say it was the last glacial maximum that, of course, affected distribution of species and genetic variation as well. Yes, and the other component of this is adaptive variation. So it means that affects specific genes, regions in the genome that are not under natural selection, and in that case, of course, they somehow influence the success of the species, yes, why it has so broad distribution range, why it covers so many different environmental gradients, yes, and there are many really examples of local adaptations, for example, in timing of bud set, when this pine species prepares for winter set in the first growing season, yes, this terminal bud, and there is nearly linear relationship between the northern European populations, like Finnish one, that set terminal bud, let's say, a few weeks earlier than the most southern populations. Yes, and there are many other examples of so-called local ecotypes of the species that maintain their unique phenotypic features. So this is all about local adaptations, so it means selection of different genotypes in different parts of the range in response to environmental factors. So this is kind of cartoon picture, yes, but if you think about this, like let's say warm and cold, and we have here different individuals, they have some variations. So on the lower panel, we will have neutral variations. So this one, which is just segregating in a population because of mutations and some neutral process. And let's assume that there is some gene here, which is important for species to adapt. Yes? So if some beneficial mutation happens, it will start increasing the frequency. So it means that you may expect for some regions that are important for adaptations to have skewed allelic frequency. So at some genomic regions, there should be some fixed alleles. And as you compare to the neutral evolving regions, there will be some just random variation. Yes? So that's the basic concept. It means that natural selection in that case will favor individuals through, of course, genes, yes, that are characterized by better growth and reproductive success. So you can consider this as a kind of fitness curve. So if you have southern populations, they will perform best in the south. But if you move them in the north, of course, their fitness will be lower and lower to the local population. So that's the basic concept of how populations adapt and what's behind if you think about the molecular basis of this adaptation. So, and this is all happening in the context of migration. On one hand, we are talking about the species that have, let's say, a good dispersal mechanism. So genes are dispersed by pollen at larger geographical areas and, of course, seeds. So on one hand, we have migration between populations and different parts of the distribution. But on the other hand, we have this selection coefficient. So it depends how about the trade-off between migration and selection. Yes, if migration is, let's say, efficient, but selection is strong, the populations will start differentiating. So the process is very dynamic and it occurs in this coexisting system of populations. And apparently it's interesting that for many forestry species, majority of genetic variation is present in every single population. So for Scots pine, about 98% of variation of the whole species is present in every population. Yes, so there is lots of variation that is available actually for adaptation and for selection. So as we know, of course, Scots pine has very extensive Eurasian distribution range, and this is important. Even though we have several hundred years, let's say, of silviculture, yes, it's not like in crop plants when you have very, very strong selection for some traits. We have some, but considering, you know, this long-lived organism with long generation time, there is still really many natural variation that was not selected for. Yes, so this selection cycle, maybe we have 3-4 maximum. And also, when we think about the genetics of the species, it's interesting that mitochondrial DNA is transmitted by seeds, chloroplast DNA by pollen, of course, nuclear DNA by both genomes. But Scots pine has well, the height of the tree is proportional to the size of the genome. Yes, it's roughly, well, 20-30 giga base of DNA, so 7 times bigger than human genome, and it makes some problems to study this species. But for the purpose of this talk, I put several questions. So first of all, I would like to show you what are the genetic relationships between populations across this large distribution range. Also, historically, yes, when we think about the past, what was happening here and what's the imprints of those processes on the genetic level, and then are there any molecular signatures of local adaptation? So this is what I showed as the example, yes, of many quantitative traits variation. The question is, okay, can we see any molecular signatures of that? So first of all, we use some next-gen approaches to develop novel mitochondrial DNA markers, which, as I said, it's kind of molecular stamp because seeds are transmitted at relatively short geographical areas, so we can better track the phylogeography of the species. So We applied 11 of these polymorphic regions, over 120 populations, and we discovered 75 new combinations, let's say, of this mtDNA variations. So you can see here like two major groups of haplotypes roughly. But most importantly is the distribution of those haplotypes in this present-day Scots Pine distribution range. So not going much into details, but you can clearly see that Finland is different even as compared to Sweden and Norway. There is a mixture of haplotypes in Poland as a result of colonizations from different routes. Western Europe is slightly different. Scotland is fixed for one mitotype, which means probably one recolonization route only. And a good thing is that at least pines from Russia did not invade this kind of country, as has completely different, let's say, mitotypes, and they were just, doing their things on that area, but only Russian Karelia somehow is similar to Finland, yes, but that's the same part of the range. So that was the picture from mitochondrial DNA. And we developed also so-called SNP array. So we look at the polymorphisms in nuclear genome through comparative transcriptome sequencing. We developed many thousands of polymorphic markers across the genome, use this variation to build this asymmetric SNP array, which means in this single hole you may genotype 50,000 polymorphic sites in a single individual. Yes, so it's very kind of high throughput technology. At the moment there are one array is built for Scots pine and close-related pine species, and the second one is specifically designed for Scots pine. So we use information from these markers to look at distribution, let's say, of this variation of nuclear genome at over 600 individuals, and that shows the population structure. So here we have Russian populations that are distinct. Here we have Finnish populations, Central Europe, Western Europe. Yes, so even, of course, you see the percentage of variation. Yes, on this PCA plot, it's not impressive, but still you can distinguish, even though the species is very wind, let's say, pollinated, you can distinguish some small, fine population structure. So the data were best explained by 5 genetic groups. So Russian populations, Finnish ones, Poland is here, Western Europe, and in Poland, in Polish mountains, we have some populations that seems to be distinct. So in general, we could say that, okay, roughly the population structure of Scots pine in Europe and Asia looks like this. So we asked the question, okay, fine. So what are the genetic relationship between those different clusters? I will not go into details about this model, but having genetic data, of course, you may calculate genetic variation, you may estimate some divergence times and so on. And we tested different models in different combinations or relationship between those populations. And the conclusion, or the best fitting model, is like this, that Russian populations are from this part of, let's say, Asia, yes? They are distinct, then Finnish populations form separate group, and there is kind of admixture between Western European and Balkan populations, and this, let's say, high diversity in Poland. So that was the best model. And also, that's interesting that if you have genetic data, you can look back in time using coalescence theory, how the variation occurred that explains your present-day data. You can go back in time and say, okay, what was the process that led to that? So in that case, we look at the effective population size, and as you can see, there is a transition in the middle Pleistocene. So over 1 million years ago, there is a signal of a bottleneck of the species, so rapid drop in population size. The same was true in last glacial period. The last glacial maximum about 20,000 years ago, we can observe this as well. And that was a bit surprising that 4,000 years ago, there is also a signal of population size reduction in this species. So the conclusion from this neutral or background genetic variation analysis is such that we can say that we have several genetic groups for Scots pine. And this is interesting, that even though the species is wind pollinated, can disperse at large, areas still those groups kind of maintain the genetic identity. Yes, so it's not like everything is mixed. So it's probably the situation that migration is possible, but selection on a local scale is strong as well. And of course, we need to think also about the phenology. Even though the pollen can be released at large geographical areas, of course, the cones needs to be receptive. Yes, so it's various and various factors like affect this signal. Yes, so we looked also. Okay, are there any signatures of local adaptations at molecular level, let's say? So in that case, we included 39 populations. This is, climatic, let's say, envelope for the species, and you think about annual mean temperature and precipitation. So those are our populations. We looked at many environmental variables, but temperature in that gradient, of course, was the major player. Yes, definitely. So using different outlier detection tests, so we look at this all variations, but wanted to find the alleles that show kind of clinal variation across the species. So we found different loci or genes behind that. This is the allelic frequency spectra, let's say, between different populations across this latitudinal gradient, which is also related to temperature. This is just example of some alleles, variation alleles in different populations. This is for Poland. Even at the small spatial scale, we observe some allelic frequency spectra that can't be explained by simple, neutral processes. So this is what we found is kind of genetic mismatch of Scots pine populations when you exactly think about the future predicted, let's say, environmental changes or temperature changes. So in general, the genetic data show that Scots pine will need to reshuffle a lot its genetic variation to really follow this environmental, let's say, niche. Yes, so in our simulations, it's at least a few hundred years. Yes, it will take for Scots pine naturally to follow up these rapid environmental changes. So that's somehow perfectly fits what Marcin said, what Daniel said as well. Yes, that we may think about the kind of assisted migration strategies, or looking at genetic data better explain why the species is suffering at the moment. So to conclude, I would say there is weak population structure in the Scots pine distribution range. That's true, but it's still population structures. The variation of outlier SNPs is highly correlated in our data set with mean temperature, and this is what I already said. Yes, that we can indicate kind of high genomic offset of a large spatial scale. It will take naturally hundreds of years for Scots pine to follow up these rapid changes. But on the other hand, the species has at least 5 million years ago went through, different glacial periods and climate changes, is still there, has the largest distribution range of all pines. So I think we should think also about this, we think it will be doing very badly in the nearest future, probably yes at some point, but on the other hand there is lots of genetic variation and potential for the species to recover. Yes, and we even didn't touch the issue of the genetic plasticity, epigenetic interactions and other things that I think will help Scots pine to manage, yes, in general. So thank you very much. That's it. I would like to endorse also some people who contributed to the research organizations, and the research was funded by National Science Center. And actually, I selected this picture because you are going there, yes? This is from Stołowe Mountains. There are several pine species growing there. We have another project, and we are sampling a lot of pines in this area. We have some results, so I'm happy to see director of this national park, because we will have this year or next year a lot to talk about this pine divergence in this area. So thank you very much.
Thank you very much, Professor Wachowiak. And now I invite for a coffee break, discuss 25 minutes. will address the questions: can we conserve forest genetic resources ex situ in Poland for future generation in face of global climate change?
Thank you. Hello, everybody. I am very happy that this is the possibility to show our research to young scientists. It was good idea, director, to collect young scientists. This is our future. So hello, everybody. Good afternoon. And it will be about seeds storage. So we have to see more about and know more about seed biology, how to conserve genetic viability, because it is mostly in seeds, gathered in seeds, genes. So seeds are collected in ex situ in gene banks. For example, like UK in Millennium Seed Bank, and in Fort Collins in National Laboratory for Genetic Resources Preservation. I will show the results for 6 oak species from this research here. And in Svalbard there is no people here, only equipment, and seeds are stored in -18 degrees of centigrade. Now crop species as a backup resources for resources which gathered people in their countries. Later will be maybe wild species. This is Polish gene bank of forest tree species situated on south-west part of Poland in the Karkonosze Mountains. Here nearby, it was ecological disaster here in 80s, so because of pollution, so the decision was to create this gene bank in this area. What about collecting of seeds? Valerie Pence from Cincinnati Zoo and Botanical Garden proposed such definition of exceptional species that we have problem to obtain recover seedlings from stored seeds. On different steps of collecting, there is a little only some seeds. We cannot collect more. There's problem. And another step, desiccation. Some seeds are not desiccation tolerant. So cryopreservation is the only way. So we have to then store it in low temperature. Some seeds are very sensitive to freezing sensitivity. Yes, for example, Fagus sylvatica tolerate -10, but not -20. And recovery, we have to know some seeds have dormancy, and in our laboratory protocols for how to break seed dormancy for 100 of species was developed during 50 years of study. So how we can define species to desiccation sensitivity? Roberts divided species for orthodox and recalcitrant seeds. Bonner orthodox seeds also manage about suborthodox species and recalcitrant species from temperate and tropical species. But this is biology. We cannot put seeds in several boxes, and Patricia Berjat, I like this, put species on the line, so from very tolerant to desiccation to very sensitive to desiccation. And even among one species, for example, it was experiment who showed this, seeds can be differently sensitive or more or less to desiccation. For example, Acer pseudoplatanus even escape from category recalcitrant in France to suborthodox. We can desiccate the seeds more. The same in Aesculus hippocastanum. So we have to know this. How we can store orthodox seeds? This is a simple method, traditional method called storage, because we can desiccate the seeds and store in cold -18. It is 0 Fahrenheit. This also it can be -20, of course. And during this storage, we want to know earlier. Before the germination collapse start to decrease, and this is we try to use epigenetic study for this, and we measured with collaboration with Professor Barciszewski and Marcin Michalak from Institute of Biochemistry, methylation of 5-methylocytosine, the level. Cytosine is methylated, and we in chromatography we measure this amount of these drops. So we can see here that this is Pyrrhus communis seed storage desiccated to 8% of moisture content. After 1 year of storage in 3 degrees, germination is okay, the same level as not stored seeds, and seedling emergence also, but on the level of DNA, we can see that amount of 5-methylocytosine increase. So the chromatin is condensed, and some genes are stopped. So some maybe proteins responsible for storage can be stopped and not produced. So this is as an indicator of first. What happened in seeds during storage. For orthodox seeds, as a backup for traditional storage is cryo storage. So we determine the safe range of moisture content. What we did, we desiccated seeds to different levels of moisture content, and we put these seeds to liquid nitrogen for 24 hours. And of course, most seeds have dormancy, so we had to seed germination. We have to remove the break dormancy. This is the control for storage in liquid nitrogen, seeds with different level of desiccation. So we determine for different tree species in this range of moisture content, we can safely store seeds in liquid nitrogen. Recalcitrant seeds, there is a lot of problem to store because they are metabolically active after shedding from the trees. They germinate during storage. And the moisture content is high. For example, acorns have 40% of moisture content. Here we did experiment when we could concentrate on individual seeds, because this is a method how we determined the germination and seeding emergence test. So we cut this part, but embryonic axis was also present on the opposite part. We cut this part, and we put acorns upside down to the soil, and this is root going down, and this is shoot, and we could count germinability and seeding emergence after strong desiccation, individual seeds, and freezing. And what happened? And this is probability how they survive -3. It is okay, -5. But this low temperature accumulate, this is for 2 weeks only storage in this. It is different after 1 month, 6 months, and 1 year, because the wrong effect of cold accumulate negatively in seeds. So -7, it depends from provenances, but what is important, desiccation, what happened, what helped during storage in low temperature for orthodox seeds doesn't work opposite reaction for recalcitrant seeds, because here is moisture content, for example, and you can see that for this low temperature, desiccation only very little helps to obtain more seedlings. And it all happens on the very low level, maybe 10% of seedling emergence, so this is not worthy to do that. And minus 18, all are dead. So I did experiments for 6 acorn species to desiccate embryonic axis isolated from this 3 acorns species belonging to 3 sections, Lobate, Quercus, and Virentes. And we can see, for example, how we do that. If we put embryonic axis without desiccation and treated by cryoprotectant ice, kill the cells because destroyed membranes in cells. But if we desiccate and treat by sucrose and glycerol, cryoprotectants, we obtain the state in the tissue, in the cell like a glass. So we can, in this seed moisture content 25%, obtain survivor. And we did some experiments for these 6 species. This is Ecology, Shumardi, Velutina, Macrocarpa, Robur, Virginiana, to determine by DSC, different scanning calorimetry, the level unfrozen water, when here we have crystallization and the death of tissue because of frost, and critical moisture content, the death because of desiccation. And this is for Ecology, this is for Shumardi, this is for Velutina. These oaks are more tolerant to desiccation. And I would like to show you Quercus robur. Here, what does it mean? Here they lose viability because of desiccation, because we have to obtain this level of moisture content to don't kill because of crystallization. So it is very difficult to put in liquid nitrogen embryonic axis. This is Quercus virginiana. This is a reaction similar to this section of oaks. And of course, we try to cryopreserve genetic resources of Quercus using somatic embryogenesis. In meanwhile, when we couldn't succeed with embryonic access, and we isolate from embryonic access this part because the radicle is more tolerant to desiccation and survive in vitro only root, but we cannot obtain plant only from root. So we use 1 millimeters in high plumules. Of course, we have to know how to culture whole plants from this plumule in vitro. So starting this step, we have to know very know about in vitro culture. And this is the moment when we started to do in vitro culture for oak. Then we cloned the monumental oaks of using in vitro culture. So this is from cryopreservation experiments it started. And this is an example of somatic embryogenesis. We obtain cryopreservation in good success, and obtain somatic embryos after cryopreservation and plants. We published these results in 2005. And this is new results about Quercus petra, because in this gene bank, we collaborate with Kostrzyca Gene Bank, a group who was working there. Urszula Basileńczyk, we published in this year the results which we obtained first for Quercus robur, for Quercus petra. There are 2 main species in Poland. So she used the cryoplates, and this method is very good because survivor is very high, 60% for Quercus. Normally, tissue of Quercus die in -9, but this tissue after cryo-pretreatment survive -196 Celsius. So this is how to use. We don't have time and isolate the plumulus. This is a cryogenic storage department established in 2019 in Kostrzyca Gene Bank. Going to the end, I would like to only show you a few slides how we clone. This is also ex situ conservation, but on the field. We preserve genetic diversity 800 years old oaks which are dying. We start from these explants. After sterilization, we put on the agar medium in vitro where we have micronutrients, the explants. We remove leaves and start to do in vitro culture. João from Brazil, who is with us and doing research proof, because we have problem with shoot tip necrosis, and he proved that BAP, that this hormone regulator, which is necessary to use in BAP, destroyed xylem, and vessels are not so strong, but when he used kinetin with BAP, it helps and we don't have necrosis. He do a very good anatomical and physiological research on this material in vitro. The new plants from in vitro, they remember their age and flowering very early, not like trees when they have 70 years old in forest, already after transplanting auto acclimatization from in vitro. So we planted, this is the oldest oak in Poland, 800 years old after in vitro culture in 2019. And they produced, this is the picture from this year. We had a little bit frost this year, but I hope that flowers survive. There will be acorns on this tree. And we plant these trees. This is a very famous oak, 800 years old. He died already because it was fired. But we had in our collection the shoots, and we recovered this tree. And it was monumental tree, and now our research can be used in forest practice because forestmen want to establish a seed plantation because there is a problem with seeds. So we collected explants from very good flowering trees, individual trees. And why is in vitro of Quercus, I should say, necessary? Because we cannot reproduce individual trees of oak using vegetative method, because it's not possible for this species. Why is in vitro culture necessary? Thank you very much. Thank you.
Thank you very much. I invite Professor Ewa Kalemba. Professor Kalemba will discuss factors determining seed viability and successful seed germination in changing world.
Good afternoon, everybody. Yes, I will talk about factors determining seed viability, and in general, the title is the research topic of all our team, which is, sorry, and the pointer is, okay, now I see. Yes, the research topic which is carried out in Department of Developmental Biology. In nature, almost each tree starts life as a seed. but to become a tree that produces its own seeds, it's a long-lasting journey. In this life cycle, seedling formation is the most critical stage for plant establishment in the climate change scenario. Successful seedling emergence depends on successful seed germination. And seed germination depends on seed viability. And seed viability depends on seed longevity, which is a polygenic trait, which is acquired during seed maturation and very important for ecological restoration. This sequence of cause and effects can be disturbed, can be affected by the process of seed aging, because seeds are prone to the aging effects. And it's obvious that aged seeds will germinate in lower percentage and will emerge less seedlings. Longevity declines with age, and then seed quality is getting worse and worse because seed protection mechanisms, seed repair mechanism, and predominantly seed detoxification mechanism are working less efficiently. And as a result, we can see a decrease of all these mechanisms, of all these three mechanisms, which makes the poor germination. Recently, a genome-wide association study identified 12 genes which are responsible for the differences in seed longevity. The 12 genes were involved in the following processes: the reactive oxygen species metabolism and the seed coat development. However, the reactive oxygen species metabolism has been considered as seed viability determinants for several decades, so it's nothing new, it's just a confirmation. That's why, at first, we will focus on the importance of the seed coat. Let's imagine the seed coat like a magic coat with packets full of superpowers. So let's see what superpowers are hidden in the seed coat. Seeds are surrounded by dead organs enclosing embryos, DOEs. And these DOEs are very important for seed dispersal in the ecosystem. But also, dead organs are involved in seed longevity. Dead organs are involved in seed germination. And also, dead organs are involved in seed establishment. But how? How dead organs can be involved in these processes? It is possible because DOEs can release hundreds of proteins just after seed hydration. And the proteins involve ROS, detoxifying enzymes, stress-responsive proteins, including defensins, and active hydrolytic enzymes, which are acting as antimicrobial substances. So let's take a look at these three molecules, which are called reactive oxygen species. And to answer the question, where do reactive oxygen species come from, we need to keep in mind that from the oxygen, which is everywhere. Because reduction of the molecular oxygen produces superoxide ion. Then this mutation of superoxide produces hydrogen peroxide. Partial reduction of hydrogen peroxide produces hydroxyl radical. And full reduction of hydrogen peroxide can produce water. Each of the three reactive oxygen species displays specific characteristics. For example, superoxide ion is the precursor molecule for many other types of reactive oxygen species. Hydrogen peroxide is the most mobile and long-lived reactive oxygen species, which is also involved in cellular signaling. And hydroxyl radical is the most reactive one. The next question is, where reactive oxygen species are produced inside the cell, inside the living cell? At first, in each organelle which contains electron transport chain, ETC, which is present in chloroplasts, mitochondria, and peroxisomes. And also reactive oxygen species are produced in all subcellular compartments where oxidases are available. It means they will be produced in cytosol, plasma membrane, and cell wall. So answering the question where ROS are produced, we need to answer everywhere. And that's why an efficient antioxidant system is needed in seeds to keep the ROS under tight control. The next question is, where reactive oxygen species will be produced inside seed cells. They will be produced during seed development, they will be produced during seed storage, and they will be produced during seed germination, because the synthesis of reactive oxygen species coexist with seed dehydration, coexist with seed hydration, coexist with the reactions like ATP synthesis, auto-oxidation, and also coexist with the degradation of storage material. So answering this question, when ROS will be produced in seeds, we have to answer that at each developmental stage in seeds. So concluding from the two last slides, reactive oxygen species will be produced every time and everywhere in seeds. That's why a balance is needed, a homeostasis between the levels of ROS and the activity of the antioxidant system. Otherwise, ROS will accumulate and the oxidative stress will be initiated. Among the seed categories that Paweł was talking about, the recalcitrant seeds will be the most affected by the climate change because recalcitrant seeds are the most exposed for the environmental changes. Also, intermediate seeds, which are called also suborthodox, will be affected by the climate change, but to a less extent than the recalcitrant seeds. As you can see in the scheme, we can see in forest many tree species which produces orthodox, intermediate, and recalcitrant seeds. In our research, we focus on 3 species which are producing recalcitrant and intermediate seeds because they are problematic for long-term storage, and of course because they will be affected by the climate change. One of the species that we work on is the European beech. European beech produces seeds irregularly in up to 10 years intervals. That's why there is a need for long-term storage of seeds for forestation activities. European beech are classified as intermediate or suborthodox just because they lose viability faster than typical orthodox seeds. In embryonic axis of beech seeds during storage, we localized reactive oxygen species. We localized them in embryonic axis, and they were present at the apical part of the embryonic axis, the part which overlaps with the root apical meristem. And the interesting thing is that the area where ROS were produced and accumulated was enlarging as the time of storage was prolonged. We also quantified reactive oxygen species, and we measured them and demonstrated that they accumulated with seed age and contributed to the decrease of seed viability. We also provided evidence that the accumulated ROS contributed to the oxidation and damage of membranes, and we also provided evidences that accumulated ROS can contribute to the oxidation of proteins by introducing carbonyls or by oxidizing the methionine amino acid. Then we focus on mitochondria, morphology and ultrastructure, and we observed that in aged seeds, after long-term storage, the mitochondria are smaller and are not present in large aggregates which were observed in short-time storage seeds. Also, we observed that the amount of crystam, the inner membrane of mitochondria, was reduced in aged seeds. So, knowing that the structure of mitochondria deteriorates with age, we performed also the analysis of mitochondrial activity using the seahorse analyzer, and as a result, we demonstrated the gradual decrease of respiratory conditions in beech seeds during storage because the oxygen consumption rate was decreasing as the storage time was prolonged. For more details about the seahorse analyzer and mitochondria, please find and ask Hania and Evelina, the mitochondria experts. Now we move on. Because in aging beech seeds, we also performed proteomic analysis, and we were able to identify proteins which were responsible for the membrane hypothesis of aging. And in particular, we identified the proteins which are responsible for the damage of the mitochondrial inner membrane. Also, with the proteomic analysis, we were able to underline the importance of the oxidative modification of methionine in proteins, which is acting in seed longevity. Because among all identified proteins which contained the methionine sulfoxide, we identified over 10 proteins which were previously assigned in the articles like modulators, indicators or markers of seed viability or seed longevity. And our result demonstrated that all of these proteins were oxidized. It means that they were not active. Another species that we work on is the pedunculate oak. And pedunculate oak produces recalcitrant seeds which are desiccation sensitive, and irregular seed set in this species and also the recalcitrant phenotype of its seeds encouraged us to search for a new protocol for a longer storage at lower temperatures than the recommended one. We assessed the effect of the storage temperature, the -3, which is the recommended one, and lower, -5, -7, and also a combination of storage temperatures because seeds were transferred from -3 to -5 or from -3 through -5 to -7. So we assessed the effect of this temperature on the accumulation of reactive oxygen species and seed viability. We measured the 3 types of reactive oxygen species, and only for the content of the hydroxyl radical, we demonstrated that it was correlated with all the seed viability determinants that we measured. And as a conclusion, we suggested that the measurement of the quantity of hydroxyl radical might be a new indicator of the seed quality in pedunculate oak. The problem is that at -7, the number of non-germinated seeds is increasing, and it is at least doubled. And the second problem is that at -7, the number of fully emerged seeds into seedlings is reduced, and it is at least half depending on the provenance. It means that at -7, pedunculate oak seeds deteriorate faster than at -3. To find out what molecules are responsible for that, we performed metabolomic analysis in which we demonstrated which metabolites are responsible for seed deterioration. In general, we demonstrated that seeds were unable to initiate the pro-survival mechanisms because seeds were experiencing freezing stress. Also, seeds were displaying declined antioxidative potential, and they were unable to reprogram the seed synthesis of energy pathways. Among all metabolites that we identified, we selected one, BTO, because this one metabolite is also involved in the synthesis of reactive oxygen species, and the level of this metabolite increased in seeds which were characterized by the lowered germinability at -7. And we suggested that the level of this BTO, of this metabolite, might be a new indicator of the reduced quality of the pedunculate oak seeds. For more details about oaks, like you heard before, please find and ask Paweł Chmielarz. And one conclusion, one perspective for the future, we will never stop studying the effects of ROS on seed viability. Year by year, we have new tools that we will continue our work. So thank you for your attention.
Thank you very much, Professor Kalemba. Now I invite Professor Tomasz Pawłowski. Professor Pawłowski will explore the impact of climate legacy on the seeds and seedling trials and European beech populations.
what we observe, the changing of environmental conditions might alter the biodemographic dynamics of the populations of species distribution ranges. Okay. I am Tora. Thank you. So, especially at the northern edges of the three species ranges in Europe, the increase in temperature is expected to broaden the sustainable niche for some species. However, direct genetic adaptation to the specific local habitats may reduce such expectancy. Environmental change may reduce disturbances in tree regeneration if future climatic conditions do not match the historical imprinted adaptation requirements for seed germination and seed fitness. As various species may be differently affected by those changes, the effect will be reflected in composition and structure of the future forest. Beech forests are mainly developed by natural regeneration, and ecotypes have developed that are adapted to the local climatic condition. It shows the ecotypic nature of variation growth traits, whereas the clinary variation was found for the spring phenology. However, the plasticity and the response to the response of beech population to the local condition has also been discussed. In particular, for the plasticity of seed dormancy and germination has not been sufficiently researched. Sorry, but I do not have this. So something with synchronization of my slides. There's some technician here to help me? Bo widzę coś innego, tutaj jest jakieś... Czemu nie pokazuje tego? Co jest na slajdzie tutaj?
To jest kolejny slajd, tu ma pan ten.
Okej, dobra. Dobra. Thank you. Beech is known to be drought sensitive, thus its population at the southern limit of the species distribution are the most endangered by the climate change, by the drought and or the increasing temperature. However, northeastern border of its natural range is limited by both, also cold temperature, but cold temperature and drought. There is growing concern about the future survival and sustainability of beech ecosystem throughout Europe, as beech may be unable to adapt to the future environment conditions in its current natural range. Especially population growing at the marginal sites are generally subjected to unsuitable conditions and have a lower chance of survival. Beech seeds ripen and are released in the late autumn. During winter, they lose dormancy, and in spring next year, they germinate when conditions are favorable for the seedling establishment. At the leading northern edge, the increase in temperature may increase germination success and beech regeneration, but more detailed studies are needed to elucidate the effect of climate warming on beech regeneration. The ecological niche of tree species is typically narrowed at the early then later life stage. It means especially for the germination or the growth of the young seedlings. Seed germination is dependent on environmental conditions acting during maturation. It means on the tree and also during the storage in the soil as a soil seed bank. Climate change can cause the disturbances in the seed germination if the history, what is in genes, do not match with what is now present, with the ecology. To access the adaptive potential, there is a need to recognize intraspecific genetic variation in fitness-related traits and its environmental determinants. Seeds, dormancy breaking, and timing of germination have important implications for the success of plant establishment in the face of the environmental risk factors. However, the role of seed dormancy and germination in the success of trees remains underestimated. The aim of presented here study was to determine intraspecific genetic variability among the beech population early fitness traits. It means seed dormancy depth and germination parameters in relation to the climate of the population origin. And also we do the experiment with the early growth seedling measurements in the common garden experiment. We hypothesized that variation in dormancy and germination of beech seeds is associated with climate conditions acting on the population, local climate, it means, and climatic characteristics of seed provenance affect seedling traits. The seeds were collected from beech stand representing different natural forest and climatic region in Poland near the northeastern border of its natural distribution range of beech and beech. To analyze the depth of the dormancy and germination trend, the seeds were subjected to the stratification germination times, which removed the dormancy, and the temperature which we usually use is the 3 centigrade. We measured the parameters like T0, it's a time of germination start, time of germination at the germination capacity, and T50, it means the time of necessary for 50% of seed to germinate. These traits reflect the dormancy depth. Also we measure germination capacity. It's a cumulative germination, it means maximum germination, seed viability using TTC test, and germination speed. It means the difference between TE and T0. And seed mass. These traits reflect seed quality. And we did also the nursery common garden experiments for the seedling, growth, and morphology analysis. So seedlings were measured for the growth parameters, and also the more detailed analysis, morphological analysis were done. And the climatic data was taken from the Climate EU. And any correlation between this seed germination, seed dormancy trait, and climate, and were tested, and also the correlation between seeds traits and seedling traits. And what was interesting for our experiment that because we observed the differences between seed masting for the two regions of Poland, it means north and south, because we take the seeds from the north 2019 and from the east from the south, and we observed this difference in the masting, which compare with the temperature and the precipitation average during last 10 years, and here's the average temperature, and here the precipitation. Here was this equilibrated as 0, and there are the deviations from this 10 years average temperature, and we saw that the mild winter, end of winter and cold spring, as well as the moist end of summer of the current year, but dry midsummer of the previous year were favorable for the beech seed germination. Observed differences in the beech masting between the regions of Poland may be the result of climate variability, for sure it is. But these differences were observed since 1945 by Professor Suszka, and so the masting depends on the weather in the region of Poland. Climate conditions, it means the disturbance can influence the masting, what especially is investigated by Professor Bogdziewicz. He found that now, when the climate is changing and because of the warming and drought, there is the problem with the masting. And beach population, we can conclude that beach population can suffer much more because of the especially less water availability in the year of the current year of the when the seeds were produced. This graph presents the germination parameters in the investigated population, and we found significant variation among populations, each populations which we were investigating, and also between these two regions of Poland. And the variation was also, and this variation showed that seeds from the south, they need a longer stratification, period of stratification, so the dormancy is deeper, but they have also better seeds. They have also better seeds viability, but germination speed was lower, and seed mass was also lower, and the germination capacity for the south species was also lower. And when we try to look on the association with the climatic variables, so we do not found the direct association with the temperature, average temperature, or with the precipitation, but we found that the T50 parameter, as I told, that is associated with the dormancy depth. This parameter was associated with the frost. BF. This is first graph present the beginning of frost period and with the evaporation. And for the germination capacity, so it means the maximum of the germination, we found that it correlates negatively with the temperature and moisture availability parameters. There are the graphs on the left. But positively with the precipitation. So the germination of each seeds depends on the climatic condition of the provenance and is associated with the seasonal variation of temperature and rainfall. The pattern of variation found in these parameters indicates adaptation of population to the climate of the seed origin site. Looking for the seedling growth parameters, we also found the differences between this seedling growth and also the morphology among population and also between two regions of Poland. Population from southern region had seedling with larger diameters and greater finite biomass and seedling than the seedling from another region. Looking for the correlation with the climatic variables, we found that seedling collar diameter, root length were negatively correlated with temperature and temperature variables. And also the positive correlation was found between stem diameter and shoot length and the map, the precipitation of at the local site. Overall, the pattern of the correlation indicates that better growth of seedling was associated with cooler and moister condition at the site of the seed's origin. Seedling morphology and climatic correlation analysis show that leaf mass fraction increased with the temperature and decreased with the annual precipitation. Stem mass also decreased with the temperature, and what was interesting that temperature exhibited strong positive effect on the root mass and density, but negative effect was reflected by a shorter root length and warmer sites. In the drier sites, the root length was longer. In the drier and colder, they were shorter. And this is associated with the water uptake. And the correlation between seed traits and seedling growth showed that fitness of beech correlate with shallow dormancy and with higher germination capacity and heavier seed mass. Seed traits correlation with seedling morphology showed that faster germination was associated with lower biomass allocation to the leaves, and positive correlation what found was stem mass fraction. And I can summarize this result that we indicate the existence differentiation adaptation in seeds in seedling ecology among provenances of Fagus sylvatica in Poland. This adaptation strategy is related to the environmental condition at the provenance level. Population originating from warmer and drier sites germinated later with the lower success and produced seedlings with shorter and shorter roots. They had deeper dormancy and poorer seed germination capacity and are likely more vulnerable to the environmental condition. In general, conclusion is that the climatic condition and the origin sites shape the intraspecific variation in seed germination and seedling traits and may limit regeneration from seeds and affect adaptation potential of beech to increasing temperature and decreasing precipitation. And we saw that what was not expected, that the northern population have worse parameters, and because we thought that climate change will for them be positive, but we think not, because beech is dependent on the water uptake, so this can be the factor limiting beech natural regenerations, especially on the north. Thank you very much for your attention.
Thank you very much, Professor Pawłowski. Now I invite you an hour's lunch break, and people who have presentation today, please contact Grzegorz Płóciennik and upload your presentation. 1 hour. Lunch, and now I invite Professor Maria Rudawska.
Mam tu do tego mówić, tak? I hope that everyone is here, so let's get started. Welcome everyone, good afternoon. And for the next 20 minutes, I would like to focus your attention to the absolutely fascinating subject, I mean mycorrhiza. Mycorrhiza is a mutualistic relationship between two species where both partners benefit. In mycorrhiza, we will speak about fungi and roots and plants. Mycorrhiza is extremely widespread. So sometimes we may say that most higher plants, and generally we say that most higher plants don't have roots, but they have mycorrhizas. This is not my statement. There is a professor of ecology, Began, and co-authors. So please remember, most higher plants do not have roots, they have mycorrhizas. The term mycorrhiza was first used by German researcher Albrecht Bernhard Frank in 1885 and originates from two words, Latin and Greek, fungus and fungus, fungus and rhiza root. In how it happens that the mycorrhiza has been discovered. In early 1880, Albrecht Bernhard Frank, a distinguished professor of higher agriculture school in Berlin, he was asked by the government, by Prussian government, to develop the production of truffles. You know what is truffle? Truffle is a very, very well known in many countries delicacy due to the special taste and smell. Maybe some of you tasted truffles. Sometimes some people like, some people doesn't like it. Unfortunately, Frank didn't succeed in growing truffles. Maybe it is good because, by the way, he discovered and described the essential structure and functioning of symbiotic relationship that he afterwards termed mycorrhiza. You can see here how accurate this drawing are originating from Frank, and they are very accurate observation he did, and his afterwards was able to present and publish in a very special publication in those times, Berichte der Deutschen Botanischen Gesellschaft. This paper was afterwards translated by famous professor, mycologist from US, James Trapi, and published in our flagship journal Mycorrhiza. So you can find this paper written by Frank. Frank was also an author of the first experiment showing this beneficial effect of mycorrhiza. So you can see she simply took forest soil and partly sterilized and partly not sterilized it. And you see that instead not sterilized soil full probably mycelium of mycorrhizal fungi, plants, Scots pine trees were growing much, much better. Also, the discovery of the mycorrhiza and its ubiquity is generally attributed to Frank. The claim that root fungi may be beneficial to plants has been made few years earlier, and I would like to mention it because it was Polish botanist Franciszek Kamieński, working first at the university in Warsaw. There was even not university at those time, and afterwards he moved to Odessa. Professor Kamieński was working in time when Poland was not on the map of Europe. Nevertheless, he made a very important discovery because he was the first person who found that the relationship between roots but I mean about these feeder roots, the finest root of the root system, that the relationship between fungi and roots, it's not a disease as it was thought before, but this is the relationship beneficial for both partners. So please remember about Franciszek Kamiński. What's interesting is that he described this phenomenon on the very special type of mycorrhiza. It means monotropoid mycorrhiza, where mycorrhizal fungi colonize such a chlorophyllous plant, Monotropa hypophytis. Mycorrhiza as a whole is not a homogeneous relationship between fungi and plants. There's at least 7 different types of mycorrhiza. In my lecture, I would like now to focus on ectomycorrhiza. Generally, we have, roughly speaking, such 2 types of mycorrhiza, endomycorrhiza and ectomycorrhiza. In ectomycorrhiza, fungi, they are not enter the cortical cells of root. And in endomycorrhiza, they enter cells of roots. So we are talking about ectomycorrhiza, extremely widespread among forest trees, all important economically and ecologically trees in temperate and boreal and tropical forests. You see that ectomycorrhizal, the most important ectomycorrhizal forest trees, and what is important is that ectomycorrhizal symbiosis for these trees, we say, is obligatory. It means that without mycorrhiza, those trees, they are not growing well. They are unable to grow well. So only well-growing trees, they are mycorrhizal. Some numbers of ectomycorrhiza. There is around 2,000 species of plants being ectomycorrhizal. Among them, we have gymnosperm. In gymnosperm, all pinnaceae, they are ectomycorrhizal, and they form very beautiful ectomycorrhizal structure we will see in a few seconds. Also, angiosperm are ectomycorrhizal. Concerning fungi, they are mostly Basidiomycota, Ascomycota, a few Zygomycota, 3 species, and there is an estimation that there is probably around 20,000 of species of ectomycorrhizal fungi, but up to now there is described around 5,000. The distribution of ectomycorrhiza is mostly limited the mycorrhiza is distributed mostly in the temperate boreal forests of northern hemisphere. However, as you can see here, according to the color, they appear everywhere in the world. I told you that mycorrhiza is a kind of mutualism. It means that both partners are able to benefit from this symbiosis. How it works? How does it happen? Fungi, as a chlorophyllose, they rely on external source of carbon originating from plant. The host plant supplies the fungus with carbon, and sometimes is estimated that 20% of the total carbon compounds is generated by the host to the fungus. From the other side, ectomycorrhizal fungi influence and benefit the host plant in a numerous way, mostly by delivering nutrients, nitrogen, phosphorus, potassium, and other substances, and water uptake. They help in water uptake also, but it is believed that the most important role of ectomycorrhizal fungi is protecting the host plant against root pathogens, and it is realized via different substances produced by ectomycorrhizal fungi, but also by the physical barrier which is formed by this mantle of ectomycorrhizae. The most important structure of ectomycorrhiza you can see here on this slide, and you see how under the influence of mycelium, how the roots, the feeder roots, are transformed in such a beautiful structure. On one piece of root, sometimes we may different mycorrhizas formed by different fungal species. And you see here on this picture the most important feature of ectomycorrhizae. It means fungal mantle and so-called Hartig net. Harticnet is the most important feature of ectomycorrhizae, not fungal mantle. They may be very, very poor fungal mantle, only superficial presence of hyphae, but the most important is Harticnet, because Harticnet is the interface between host plant, between tree, and mycelium, and ectomycorrhizal fungus, and here is the exchanging of the substances between these two partners. As I show you already, this ectomycorrhizal mycelium is able to transform the feeder roots into very different structure, and we call them morphotypes. And according to the different morphotypes, you can classify these morphotypes sometimes with some experience, when we have some experience, even to species, but sometimes to genera. In our department which was called before Department of Mycorrhizal Research and now Symbiotic Association, we are mostly focused on ectomycorrhizal community structure. And this ectomycorrhizal community structure is above and below ground composition and relative abundance of ectomycorrhizal fungal species. This is the main subject of our research. And to this area of research I would like now to refer. So, to describe ectomycorrhizal community structure, we may apply different methods. The oldest method is sporocarp inventories. Sometimes we say fruiting bodies instead of sporocarps. We have a different sporocarps and also it may be morphotyping. It means and afterwards molecular analysis of fungus from such a morphotype via Sanger sequencing, and in the last years also metagenomic studies. Just a few words about each method. Sporocarp inventories is a long-lasting, need several years, several visits, also during one season, and must be the very good specialist to identify fungi from sporocarps. Long hours sitting under with the microscope. What's more, there's a quite big variety of fungi, this growing above ground, this growing below ground, but also I would like to focus your attention to resapinate fungi that due to the molecular method we discovered as also mycorrhizal fungi. They form their fruiting bodies of fungi growing on the surface of some litter, soil, or pieces of wood. So, ectomycorrhizal identification, it means morphotyping. It was the first step in our research when we started to analyze ectomycorrhizal community structure. Is done according to the shape and dimension of these morphotypes. We analyze feature of the mantle surface, color of mantle, presence of some additional structure like rhizomorphs or emanating hyphae. And here, when we are talking about morphotyping, I would like to remind you Professor Tadeusz Dominik. It is a Polish mycologist. He was the first author trying to identify ectomycorrhizal community structure according to morphotyping, and even author of key to ectotrophic mycorrhiza used in certain times quite often. A great contribution to the development of anatomomorphotyping, because we base not only on morphology but also on anatomy of these morphotypes, made Professor Reinhard Agerer, German researcher. He is an author of Atlas of Ectomycorrhizae and Description of Ectomycorrhizae, very often used by us in the beginning of our study. The true revolution in the determination of ectomycorrhizal fungal community was the discovery of polymerase chain reaction, and in this case I would like to remind you Carey Banks Mullis. He together with his colleague discovered and described polymerase chain reaction, and he shared Nobel Prize in 1993. This discovery of polymerase chain reaction was a true revolution in determination, in description of ectomycorrhizal fungal community, because it much it's increased the significantly, and with this method we would be quite much more precise. Ectomycorrhiza identification using polymerase chain reaction is very fast, and after PCR amplification of ITS rDNA with fungal specific primer, we and sequencing DNA, we afterwards compare our results with dedicated databases, and we have results, and it may be done from the single morphotype, as you can see, and we start to use it very soon after the discovery and description of this method. For us, such a milestone was the participation in International Conference on Mycorrhiza in Uppsala, where before this conference we took part in the workshop and learned this method. And afterwards, very fast, we introduced this method to our research. And you can see from the single morphotype we are able to evaluate the species. The next step in identification of ectomycorrhizal fungal community was development of metagenomic studies, new generation sequencing. The advantage of this method is that we may identify in such so-called environmental samples, so a sample of soil or sample of roots or some pieces of wooden pieces, we may identify not only mycorrhizal fungi but also fungi from other trophic groups. So our knowledge about microbiome of tested stench is much wide. So, as I told you, symbiotic, saprotrophic, pathotrophic fungi in one environmental sample. Now I would like to show you some examples of our study when using all this method, but as a matter of fact, to determine the whole ectomycorrhizal fungal community structure, we have to use all three methods. The chance of mushroom appearing, our first using of molecular method and morphotyping was in forest nurseries. Here you see the examples of so-called bare root forest nurseries, where seedlings are produced with bare root system, not in containers. Such a production is very, very popular in Poland. At least 90% of production of seedlings is produced in bare root forest nurseries. And there is a very high culture of production. You see, no weeds, nothing, everything in perfect culture. So the chance of using method of sporocarp identification was null. Therefore, sometimes it happens that some fungi appear, but it's very, very, very rare. So how it looks like ectomycorrhizal community structure of seedlings reared in forest nurseries was completely unknown. You see here only a few examples when in some nurseries we could find some fungi belonging to Laccaria or Hebeloma, but only these two species, two genera. To our surprise, in ectomycorrhizal forest nurseries, we were able to identify several ectomycorrhizal fungi. You see here the number of species, at least 20 species on most very common forest trees, and in total it was 95 taxa of ectomycorrhizal fungi. We found that the structure of ectomycorrhizal fungal community is slightly different between coniferous trees and deciduous trees. You see these yellow and green points. And we published the results in very many papers, so if you are interested in this subject, you can find the list of references in the book of conference. We realized also our research on ectomycorrhizal fungal community structure in different forest ecosystems, and we did it in a very different aspect, like effect of environmental pollution. We analyzed community structure under the forest chronosequence, so depending on the age of the host plant, under the influence of tree genotypes, in forest decline. We were interested how it looks like when the forest is declining, what's happened with mycorrhiza. Under the forest management, different types in forest reserves and national park, and in very different habitats like coastal dunes, peatlands, microhabitats, and so on, and so on, and so on. And for our analysis, we analyzed different host plants like native tree species you see here, but also alien tree species like Pinus nigra, Abies grandis, Caria, Quercus rubra. Some examples from our research showing the relative abundance or simply diversity of ectomycorrhizal fungi. I would like to show you this picture because you see here how it looks like the ectomycorrhizal fungal community. Here you see the very typical pattern of ectomycorrhiza. 1, 2, 3 species quite abundant, and the rest in a very low abundance. This is a typical pattern of ectomycorrhizal fungal community. Here, in this research, we for the first time determined Lactarius rufus. It was our first discovery using molecular method, so I have a sentiment to this result. We also analyzed ectomycorrhizal fungal communities in Scots pine chronosequence from 7 years to 87, and we found that there is no difference in species richness, but there is a change in the species composition. And you see here that there is a change from the in the special type of mycorrhizal fungi Suloid with abundant rhizomorphs and extramatrical mycelium into the Russuloid fungi, which completely different structure of morphotypes. Some results about fungal communities regenerating, growing on different microhabitats. Here, forest ground, decaying stumps or erratic boulders, it means such a big stones. And also we were able to show that the ectomycorrhizal fungal community was completely different depending on the substrate. So one of our last project was the comparison between forest reserves and managed forest. We hypothesized that managed forests will be characterized by much lower diversity of ectomycorrhizal fungi due to the management, but it was absolutely reverse, and we found that both managed forests and forest reserves, they have a comparable number of species richness. Species richness was comparable. However, during the several years of our observation, always managed forests, they had higher species richness than forest reserves. This was really surprise from these studies. You can also see that managed forests and forest reserves, they have several species in common, but each type of management contribute to the special number, certain number of species characteristic only for this type of management. This slide also show you that depending on the method we analyzed this via molecular analysis of ectomycorrhizas, but also by inventory of sporocarps. Also, each method contribute to the special number separate fungal species, and only 50 were in common. Not going into detail, we found that both forest reserves in managed forests, they contribute. They have a significant effect on the ectomycorrhizal community structure, but it appears that the site is more influential than the type of management. This was a rather surprise for us. When we have the detailed results of ectomycorrhizal fungal community, we may correlate these results with different environmental factors, and by that we may say which environmental factors, the different abiotic and biotic, which in the highest way structure this ectomycorrhizal community, which are the most important driver of ectomycorrhizas. And at the end of this study, we compared 3 different methods: sporocarp inventory, ectomycorrhizas, analysis of ectomycorrhizas by Sanger sequencing, and also analysis via new generation sequencing. And also you see that each method contributes to the special number of species. So only integrating all 3 methods give us the real picture of the ectomycorrhizal fungal community. Of course, we're always approaching to the total number, but nevertheless, different methods have to be used. These results from the comparison of reserves and managed forests we published in several papers. You may also find it as a reference in my lecture presented in book, conference book. And at the end, I would like to show you the most important challenges in ectomycorrhizal studies. So still new development of new methods, long-term field observation are extremely important, looking for different ecosystem, man-made, native, disturbed, virgin, also different geographic area, host trees and fungal groups are important, applying the state-of-the-art molecular tools, and development of mycosilviculture. The knowledge about fungal ectomycorrhizal community structure may be extremely useful in some development, for example, inocula for inoculation in such a type of seedling production, different completely than barroot forest nursery. It means such a containerized type. So when we know which fungi are able to form ectomycorrhizas in such a young stage, we can make advice to the producer of inoculum. I am not in favor very much of this artificial inoculation. Nevertheless, in such a condition of this containerized production, sometimes it is necessary. So, it's end. I've made probably too long. Thank you for your attention. And due to the shortage of time, I could only very briefly outline the research on mycorrhiza, but I encourage you to take an interest to this topic, and remember always, we do not have roots, we have mycorrhiza. So when you plan experiments, you have always remember that these trees, these plants, these seedlings are mycorrhiza.
Thank you very much. Oh. I invite Professor Marcin Pietras. Professor Pietras will present the biogeography of non-pathogenic fungi in Europe.
So at the beginning, thank you, Professor Rudawska, for the introduction to my topic, because all my life I'm 100% ectomycorrhizal guy. But today I would like to tell you about 10 years of my work in the institute, and I'm gonna show you how we gather mycorrhizal or ecological data, how we can proceed with them, and the result of our findings from last years. So first of all, I want to tell you what is biogeography, and the very simple, biogeography can be described as discipline dealing with the pattern of geographical distribution of different organisms and factors that determine those patterns. And here you see very simple map, and I think that we can tell that the map is a basic tool in biogeographical studies. And according to the literature, the first biogeographical map was published at the beginning of 19th century by Lamarck and Condol in the Flora of France. And actually, I tried to find what is on this map, and it's really difficult because I think that here there are some regions with high endemism marked on this map of France. But what about the first distributional map of the non-native fungus? And I think that the first one was maybe not a map, but a plan showing the distribution and spread of stinkhorn octopus, Clatrus archeri, the fungus non-native to Europe that came from Australia and New Zealand. And this map described the distribution of this fungus in Schwarzwald mountains in Germany, and spread between 1920 and 1940. All thing is that the question is that we can use fungi as a model organism, and most of the mycologists tell you that this is a sick idea. It's no way to do this. Why? Because fungi are usually cryptic organisms and forming ephemeral fluid bodies and mainly existing as a mycelium or growing the soil. Second thing is the species concepts of fungi is still unclear. And here's the example of the Rhizopogon pseudoroseolus. In the picture you see the fluid bodies of sporocarps of this fungus, which has distinguished from the Rhizopogon roseolus group with 31 other species. So these fluid bodies I found for the first time in Australia, and to confirm the identification of this Rhizopogon pseudoroseolus, I used molecular methods. And here you see the clustering of the two sister species. Here is pseudoroseolus cluster, and here is the roseolus cluster. So molecular tools give the answer and give us possibility to distinguish a whole group that formerly was classified as one species. And the last thing is that fungi, some taxa can be described for the first time or known only from invasive range. And here is the example of dead camp, Amanita phalloides. And here you see the picture showing the fungus described in mid-80s. In mid 18th century in North America. Here the same fungus in Europe. Here is the picture from the original description with diagnosis of the species published in 1916, so before the first records has been described. And the last one is a mushroom currently recognized as Amanita phalloides for both North America and Europe. But maybe we can use this fungi as a model organism, because maybe some fungal taxa can be highly suitable for invasion ecology research. And if we have available occurrence data, and here you see almost 500 records of these four species that I gathered for the last 10 years. if these fungi have distinct morphological features, making possible to identify them in the field condition even by naked eyes, and they are frequently used in invasion range. So maybe we can use this fungi as model organism. And 1 month ago, the paper has been published, where authors suggest that all Suillus species can be a model organism not only for biogeography, but also for ecology and evolution. So what we actually know about the introductions of non-pathogenic fungi in Europe. Here's the paper published in 2009, showing the global pattern of ectomycorrhizal introduction. And you see the hotspot of ectomycorrhizal introduction in South America, South Africa, New Zealand, and Australia. And Central Europe, you see that there is a blank page on the map of introduction. So, the rate of introduction depends on the group of fungi, and most of the introduction are related with Suillus and Rhizopogon, which are called Suilloid fungi. And this group altogether is more than 200 introductions and 40 species that have been worldwide introduced into new area. The question is that the number of species and number of introductions is high. That time it was quite impressive, but when we focus on the one geographic region, and here we jump to the New Zealand and Australia from Europe, the rate of introduction can be much higher. And with my PhD student Tomek Sobczak, we evaluate the number of introduction and number of species that has been co-introduced to New Zealand and Australia, using different source of data. It was scientific papers, open databases, grey literature, as well as citizen science databases. And we found that here, only in New Zealand and in Australia, we found 32 only Suilloid fungi with more than 800 introduction in this region. So it's quite a lot. But just like I told you before, this region is a hotspot of ectomycorrhizal introduction because of co-introduction with the North American and European conifers. Here one of the Suilloid fungi, Suillus lacae. We gathered distribution data from the native range and from the invasion range. And as you see, I have also a dog, so why can I prepare the model for this fungus? And what you see on the model that we prepared using Maxent, here in native range, the predicted distribution fits quite well with the known occurrence of the fungus. You see the high prediction for the occurrence of the fungus in South America and New Zealand, where ectomycorrhizal partner of the Suillus lacae, Douglas fir, is invasive species, and you can also see the high prediction in Australia and Tasmania, where the fungus can be invasive. In Europe, you see the prediction is quite high, but the limiting factor is the presence of the ectomycorrhizal partner, the Douglas fir. Here's the second example of the fungus that spread around Europe. It's It's Auroboletus projectellus, a North American bolete that has been reported for the first time in Lithuania in 2007, and since that time this fungus spread across the Europe, and this uncommon type of introduction, because this fungus jumped to the European pine species. This is how this fungus looks like. Here you see the ornamental stem, which is very characteristic for the species, and the typical habitat for the fungus, the acidophilus dry forest in Pomerania. And this fungus was classified as invasive, but because of undiscovered impact on native ecosystem, it rather should be called potentially invasive species. However, we also prepared the model for future spreading of the fungus, and on the light gray you see the potential distribution of the fungus based on climatic variables and the presence of the host. So it was two Pinus mugo and Pinus sylvestris. You see that the expansion in Europe is unstoppable. And after that, we focus on the distribution of the fungus in Poland. Here you see the concentration of the fungus in Pomerania, where was for the first time found in Poland. And here we use my own observation data from citizen science database Fungi.pl, GBIF data, records from the list of the alien species published by Institute of Nature Protection, and also Facebook. So it's also citizen science data. And here you see the number of records marked by the dot. And if we think that here the spreading of the fungus is quite natural, maybe because of spores, but we don't know, or maybe because of animal vector, but I was wondering what is the factor that is responsible for the long distance dispersal of the fungus in Poland. Because from this to here it's more or less 700 kilometers, and there is only one suspected it's human. By the way, let me introduce Dr. Paweł Horodecki, who is going with us to our trip this weekend to Park Narodowy Gór Stołowych. And for all of you who came here abroad, you should know that Poland is a perfect model country to study ethnomycology, because we are most mycophilic nationality on earth. In Poland, we use more than 100 taxa. Some of them are even edible. And in this paper, we published that we found 2 new taxa of fungi for Polish mycobiota that was sold in the open air market in southern Poland. So they have never been found in the nature, but we found it on the open air market. But the question is how to measure human vector, or how to assess the human vector. And here we use some tools that we use mainly in ethnomycological surveys. And we perform interviews in the field. My dear colleagues, Magda Terlecka and Dominika Robak, who both girls are here, so they stalked the the tourists last year on the Polish seaside and interviews them and ask a lot of questions. The second way was that we prepared the Google form and send it to all of the amateur Facebook group dealing with fungi. We ask about a lot of things that could be usable for our study, but each record needs to be proved by the photo of the fungus. And my first idea was to link the number of people who went from their place to the Polish seaside to pick some mushrooms. I know that seems strange, but we are in Poland. We are the most mycophilic country in the world. So, but it was not so easy to find the connection. Anyway, during this Google form, we found almost 50 new localities of the fungus in Poland. So I think that this is the conclusion from this research is that the distribution of the fungus in Poland is still underestimated. And second thing, that we can use citizen science tool to investigate biogeography of macrofungi. And the last fungus that I would like to tell you about is the Eclatus archeri, the same fungus from the first map of the macrofungi. And this story starts, I don't know, maybe 10 years ago, when during my PhD we found the one fruit body of this fungus in central Poland, and I gathered data from Poland. And here you see new localities in every 10 years since 1973 till 2014. We see the increasing number of localization and split from the southeast to east-north direction. And after a few years, I gathered the data from the whole world. It was, I don't know, more than 300 records from native range, so Australia, New Zealand, from unknown range, because South Africa, we don't know actually how this fungus get to the South Africa and in Europe. And we also prepared some model, and the aim of this study was to see the past distribution of the fungus. And here we used data from last glacial maximum. So here you see how the Australia and New Zealand looked like 20,000 years ago. But the most important is that you see that the highly suitable region are comparable with the known distribution of the fungus in the presence. Second thing was to evaluate where the place with highly suitable for the fungus we can find in Europe. And the last thing was that we predict the future changes of distribution of the fungus. And here the most important result is that you have see the red color here. So you have the contraction of the fungus in native range, especially in southeast Australia. The fungus will keep their niche in the New Zealand. But in Europe, you see that based on the different climatic scenarios, there will be reduction of the fungus in Central Europe, but the fungus will spread in northeast. So the conclusion from this is that the fungus can be treated in native range, but in Europe can become a invasive taxon. And few words about the early detection of fungi, because Professor Dawka told you about next generation sequencing method, and here we can use this method for early detection of alien fungi. And here's the example of few Douglas fir trees growing somewhere in beech-dominated forest in Pomerania. And during this approach, we found 4 North American taxa that was present mycelium was present in the soil. And as we know, these 3 first are psuloid fungi that are regarded as the drivers of pine invasion. And the last thing is that impact. Ian Dickey summarized the impact of invasive species, but most of these studies were conducted in South America and New Zealand. And in this paper, there's some proposal of group of fungi that can be regarded as invasive. And in my work, I launched the project, and now I'm switching from the regular biogeographical study to ecological study. This year we are finishing the 3 years experiment when we want to prove the invasiveness of the non-pathogenic fungi using different methodological approach. Including NGS. We designed specific primers to identify the fungi directly from the soil. We also assessed the live decomposition rate in the place that these fungi are present, and analyzed the enzymatic activity of soil and ectomycorrhizas. And thank you for your attention.
Thank you very much, Professor Pietras. Now I invite Professor Grzegorz Iszkuło. Professor Iszkuło will answer the questions. What the point of toothed leaves?
Thank you very much. Why this topic for you? And I think you are a young scientist and you don't have enough money, don't you? Me either. So this research is did without money, just computer, internet. So I think everyone can do this. So maybe this is one of the reasons why I would like to present this topic. And of course, this research we did very long time, and I think I wouldn't do this without young scientists, without my colleagues. So thank you, Magda, thank you, Łukasz. They are here, and I think this is very important to connect maybe some experience and energy of young people. And I think next important thing, these results were have just published. So this is a completely new thing. Okay. So relationship between percentage of untoothed species and toothed species and climate was discovered above 100 years ago, so quite a long time ago. And of course, it was later quantified. So you can see quite nice relationship between mean annual temperature and percent of untoothed species. This relationship is very important because people use it to estimate climate temperature in the past. So on the base of fossil floras, people estimate temperature in the past. So it's quite commonly used now by paleobotanists. And first of all, why do leaves have teeth? So there are many different hypotheses, but I would like to say about 3 of them. So first is that teeth imitate feeding by insects. So other insects avoid this leaf. So maybe this is piece of true. Next hypothesis, this is bud pecking hypothesis, is quite new. So teeth inside dormant buds fold more compactly leaves. So another name of this hypothesis is origami hypothesis. So this hypothesis is connected with our relationship because longer dormancy period, more toothed plants. Next hypothesis is also connected with this relationship because the name is gas exchange hypothesis. So this is connected with phenomenon that on the edge of leaf there is much more intensive photosynthesis. So shorter growing season, greater need for intensive assimilation. And we're going back to our relationship from 2011, and you can see that at these points we can't notice this positive relationship. In fact, there is no relationship. So maybe the reason is that in colder areas there are not too many localities. So let's improve this relationship. Let's improve leaf margin analysis method to better explanation of climate using relationship between temperature and untoothed leaves. The perfect area was in Finland, because in Finland temperature range is between -2.3 and 4.6. So the country is divided on 10 per 10 kilometers squares. Very similar situation is in Germany and Poland. So we could use list of species for every single square, and it was really easy to analyze this data. The question is, of course, what is toothed and untoothed leaf, and division is very easy because, of course, dentate, serrate, crenate, so Polish name is piłkowane, ząbkowane I karbowane. There are just, toothed. So this is division on toothed and untoothed species. The most difficult was estimation species. What is toothed and untoothed species, and we used herbarium materials. So it was the only one method. So every single species had to be checked in the internet. Okay, this is the result of our work only from Finland, and you can see very nice relationship. and we added Poland and Germany. Well, it's okay. But the problem is that this is a negative relationship. So the first thought was what went wrong. So we checked it double many times, but no change. The second thought was, okay, it is what it is. Let's do something. But the last was, okay, this is very nice. This is a kind of revolution. Let's change science. And the question is why our relationship is invert. Maybe this is a reason of woody species, woody evergreen species. They hibernate under snow, so low winter temperature, it's not very dangerous for them. And it's important to know that evergreen species are mostly untoothed. So above 80% of evergreen species are untoothed. Okay, first results was very optimistic. You can see increase of number of deciduous species with mean temperature, but evergreen species did increase. So it's more or less the number of evergreen species is more or less the same. But when we remove this evergreen species, this relationship is still significant, is still high. So this is not answer of our question. So you can see that we don't know why we found negative relationship. In fact, we don't know why this relationship is positive in warmer climate. So I think this is an open question, maybe for you. And we recommend don't use this method to estimate temperature in the past below 5 degrees. So our results also undermine gas exchange and backpacking hypothesis. So as I said, we don't know why we observe this kind of relationship. Thank you very much.
Thank you very much. We listened to all the lectures, very interesting lectures. Thank you very much to all speakers. And now I invite you for coffee break and discuss. Thank you very much.
4. So we should start. Good afternoon. My name is Leszek Karliński, and it's my pleasure to welcome you to this session today. In the plan we have 5 presentations. Each presentation shouldn't exceed 50 minutes. The time for questions and discussion will be provided after the last presentation. So I obtained information that the first participant, Mr. Rotowa, couldn't attend to this meeting. So, but he sent his presentation via email. So maybe for the first will be maybe I will start. Oh, okay. So, please.
The title of my presentation is Revolutionizing Nursery and Garden Practices, Peat-Free Substrates and Novel Fertilization as Strategies for Sustainable Seedling Production. Dr. James Rotowa is my name. I'm from the University of Agriculture in Krakow. Here is the presentation outline. And first, I'd like to talk about peat. Peat has been identified as a major component for substrates used in nursery, that's forest nursery, and also in horticultural gardens. The acceptance it has gained is because of its perfect physical, chemical, and biological properties, and of course its very high water retention capacity. And considering the utilization of peat, in 2012, Gruda reported that about 90 million meter cube of peatland is often excavated, and of course this emit about 2,800 years of carbon rot. This is virtually more than all the forest in the world. And when peatland is excavated, it often spread on plantation, and the aftermath is that it turns into carbon dioxide, totaling into greenhouse gas level, and also a very big threat to our treasured ecological system. From the far left, of course, we could see a green land, and within the middle picture shows a kind of exercise of excavating the peatland, and the far right shows that some kind of this is the same situation after like 7 to 8 decades. It's virtually impossible to retrieve peatland after excavation. So looking at what the future is going to be, the utilization of peat has to stop, then it's important to seek an alternative. If peatland will be conserved and no longer destroyed, then the need to survive beyond the utilization of peat has come to play and of course a force to reckon with. For us in the University of Agriculture, we came out with a concept of taking from the forest and also returning into the forest, and of course beyond the forest, in a bigger concept that addresses the innovative technology for the production of substrate and fertilizer produced from indigenous resources for the production of forestry seedlings in what we call sub-Narcont Poland. And the projects try to consider some kind of technological line chain for the production of novel substrate and novel liquid fertilizer. And quite interesting, as you can see from the images, we have some level of success from this concept, and this therefore gives rise to the objective of this report, this presentation, which seeks to consider what happened, the effect of peat-free organic substrate and fertilizer, which was developed at the University of Agriculture in Krakow, to the allocation of nutrients in different organs of tree seedlings. And the hypothesis put forward here did assume that this innovative concept will, increase the allocation of nutrients in different organs of tree seedlings. Table 1 here shows the component of organic peat substrate, and you could see that the combination of the substrate, sawdust, wood chips, straw, wood bark, and of course, all of these are materials gotten from the forest. And the interesting thing about these materials is that they all are readily available, of course, cost effective, and most importantly, they all are environmentally friendly. So a different ratio of combination, we developed 3 different substrates, which we coded here as R20, R21 and R22. Meanwhile, we have SC as the control. I haven't done this in different combinations. We try to evaluate the properties of the organic substrate, and of course they have also exhibited different level, different level of water holding capacity, and of course the bulk density and porosity also evaluated. Table 3 here shows the content of nutrients in the substrate, of course before and after sowing. And when, of course, like I said, we have to fertilizer. When the novel fertilization, which is of course liquid in nature, is mixed up with the substrate, that's when we have what we have coded here as UR. So the U concept represent the university novel concept, and the R represent the solid fertilization, which is of course the standard that is often used by state forest. The experiment was laid in a 2x2x4 experimental design consisting of 2 different tree species, that's beech and oak, and 2 different fertilization, that's forest and university. Remember, I did mention that the forest is the solid and the university fertilization is the liquid. And we have 4 different substrates, including peat, which serves as the control substrate in both fertilization. A series of data were collected from the seedling samples, but the concentration of the attention is on the far left of this table, which is allocation of basic element. These elements were considered in different organs, the stable and of course the unstable organs of the tree seedlings. At the end of the production season in the nursery, destructive sampling method was adopted, where we have the seedlings. We try to separate the root component, the stem component, and the leaf. Haven't done this. The different parts were further processed into powder form. They were blended, dried, and processed into powder. And then we have every of these packed as shown in this figure, the leaf sample, the stem samples, and of course the root samples. And standard procedure followed to analyze these samples, and the analysis was taken in the laboratory of forest environment, geochemistry, and land intended for reclamation in the Department of Ecology and Silviculture, University of Agriculture in Krakow. From the result, the figure here shows the biomass allocation in different parts of the organs, and from the first figure shows the biomass of leaf allocation. And quite interesting, when we look at oak, for example, there was no significant difference among all treatment, and also the same thing was observed in beech, except for the control substrate, which tends to be higher than others. And when we look at the result of the second graph of shoots, that's a stem, we discovered that there was no significant, not only among species, but even between species, as the treatment practically have the same alphabet. However, something interesting happened when we look at the root biomass. It was obvious that the biomass allocation in oak was significantly higher than that of beech. However, even within the treatment, we could observe some level of significant variation, not only between species but also within treatment. And the result on the next table, that this is showing the variation in oak now. Like we see in the previous graph, more allocation were in the root and the stem than the leaf, respectively. However, there was no significant difference in major elements like nitrogen and phosphorus when we look at the situation with the root. However, it was of course significant across the shoots and also across the leaves. When we look at the situation with Fagus vertica, it's quite interesting to also see that nitrogen was also not significant, and even carbon and few other elements like magnesium and sulfur, right? But anyway, just like we have in oak, there was significant difference across the shoot, the treatment, and also the unstable part of the organs of the leaves. The result of principal component analysis shows the allocation of nutrient in the root of the different species. From the upper far left, it shows that all the elements, of course, we are somehow on the positive side or on the negative side, but the crucial one for development of seedlings, which is element like nutrient like nitrogen and phosphorus, we are on the negative side when we look at figure 2 and also in figure 3, which shows substrate situation. But when we now come to figure 4, it's obvious that this element, the position changes from the negative side to the positive, which therefore interprets that the major factor of variation that we have seen in the earlier results and tables is as a result of the fertilization. Fertilization is responsible majorly for the variation among the substrate, because the substrate shows no variation, but the addition of fertilizer improves the concentration of this major nutrient in the substrate treatment. And of course, the results of correlation analysis show across board that there is high positive correlation among virtually all the elements in both species. And now, based on the results of the study, it can be said that the novel substrate and fertilizer that was developed by the University of Agriculture in Krakow has shown a strong competitiveness with organic peat substrate because they met the existing characteristics of those that have been used in the state forest and have resulted in an efficient distribution of nutrients across different organs of tree seedlings. And obviously, the success of these findings have a long-time benefit for peatland conservation effort, all right, and also it will contribute to the development of sustainable forest and garden practice, not only in Poland, but across the whole continent of Europe. Thank you very much for listening.
Okay, thank you for this presentation, and now let's move to Himalaya mountains, and I invite Dr. Łukasz Walas, who will talk about biodiversity indicators in developing forest management and eco-restoration strategies. Please, the stage is yours.
Złączony. Od początku, dobra. Okay. So, why the Himalaya mountains? Because this mountain chain have very high biodiversity, extraordinary high, as you can see on this map, which is also a result of our research. This research collect data about occurrence of plant species in Indian subcontinent. You can see that some places like Tibetan highlands and Thar desert in Pakistan have very low biodiversity, but extraordinary high is in this mountain belt from the Pakistan through northern India, Nepal, Bhutan to the Yunnan in China. And this region is also quite wild, separate the hot valley of Ganges and Brahmaputra in India from the barren land in the Tibet. Conservation efforts are, of course, very intensive in this area because of this high biodiversity. However, still this region is not very well known. According to the Worldwide Fund for Nature division to the ecoregions, we have a lot of forest types in this mountain belt of Himalaya, which is also connected with very diverse environmental variables in this area. The mountain regions are diverse because of this altitudinal range, and because of this also the forests in this region can be divided into three main groups. First are the tropical evergreen rainforests that are in the lowest elevation. Higher we have temperate mixed forest, and in the highest areas, we have coniferous and deciduous trees. Above the forest line, we have, of course, alpine zone. We focused on the one region that is on the northwestern part of the Himalaya. This is Kashmir and Jammu and Kashmir. You can see that in the middle of this area we have some valley. This is Kashmir Valley, which is around this valley we have the mountains of Himalaya. This unique bioregion exhibits diverse environmental variables, and we have here various forest types. We use more detailed division to the forest types than Worldwide Fund for Nature. Thus, we have here 12 forest types, which we can divide into 3 altitudinal belts. On the next slide, we have explanation which acronym is connected with which forest type. And these forests are from the most lowland to the highest. As we can see, also we have some the most important significant species in each forest type. I will remember this acronym for the next slides. Here we have some photos for each forest type. It's not very visible here because the colors are are not very bright. And I see that one of the photos was multiplied for some reason. Anyway, we have here lowland forests and forests from middle elevation. As we can see, this forest, for example, subtropical broadleaf hill forests are not very dense. This is quite open forest type. Also in the highest elevation, we can see that the forest types are very diverse, from the lowest Himalayan pine forest to the subalpine forests that are on the highest mountains. We collect data from each of the forest types from different parts of the Kashmir Valley, and we have 96 plots for trees, twice plots for shrubs, totally 192, and 480 plots for herbs. According to collected data about occurrence of each species, we prepared some analysis for comparison of each forest type and to recognize which species are most significant to recognize which type is which. And you can see that we use a lot of different multivariate analyses like twist-span, canonical correspond analysis, or indicator species analysis. The most significant results, we found 169 species distributed among 146 genera, and I can tell you that this is the most detailed collection of data about occurrence of plants from this region. So this is something new also for the Kashmir. Some of the species was not observed earlier. We described 101 herbs species, 34 tree species, and 27 shrub species. As you can see on these colorful plots, the colorful plots show us the Shannon and Simpson indices for diversity, and the highest diversity was observed in the Khyr Sisu forest and subtropical broadleaf hill forest, that are forest types that from lowlands. In contrast, subtropical pine forest and Sivalik pine forest have substantially lower diversity indices. According to occurrence of species, we was able to divide the forest types into 3 main groups. And these groups also, what is not surprising, are connected with elevation belt. For first group, the most significant species was Mallotus philippensis, Parthenium hysterophorus, and Adiantum venustum. For second group, Fragaria nubicola, Diopteris roboteum, and Pteris cretica. And for third group, Viburnum glandiflorum, Viola odorata, and Rumex nepalensis. Also, we was able to connect species with particular forest type. And as you can see on this slide, for forest from similar altitude, the most significant species are quite also similar. For example, we have Caricia spinarum in the lowland forest, and in the these forests that are in the highest elevation, the species are even the same. I mean, Berberis lithium, Sarococca saligna were common indicator species for all the forest type in the highest elevation belt. We also compare these forest types, divided them into the group according to the phylogeny, because when we check it without taking into attention phylogeny, we can group quite logical our 12 forest types into 3 elevation belts. But when we add this additional variable, which is the phylogenetic relation between particular species, we can observe that some forests from the lowland start to be more similar to the forests from higher elevation. This is some indication that some species from lower part of Himalaya have some counterpart in the higher elevation that belong to the same genera or the same family. On this quite messy plot from the canonical correspondence analysis, we can see relation between species occurrence and environmental variables. And we can tell that 3 environmental variables was the most important and most significant in our model. And it was altitude, slope, and grazing. First 2 are not very surprising because altitude is connected with different environment, so have strong influence on species occurrence. Slope also because on the very sharp slope, the species like tree cannot grow. But for me, a little bit surprising was the strong influence of grazing to the recognizing species occurrence. Maybe it's just underestimation of animal influence on the species occurrence. But it seems that this is a very important factor in this mountainous region. Soil erosion, cutting, and fire also was quite important, but not statistically significant for this model. And because we have not too much time, I will go to conclusion. And the most important result of our work was description of the biggest set of species from this region, from Kashmir region. This is 152 recognized species, which contribute to this botanical knowledge of the Himalaya region. The overall richness of seed plants have peaked with the intermediate elevation between 1,500 to 3,000 meters, which for Himalaya mean the intermediate elevation belt. Although the difference between forest types may indicate the effect of climate or another environmental variables, geographical distance was still important. As you can, maybe you don't remember these previous slides, but this division into forest ecoregions make some gradient from west to east, because we have some also gradient of species occurrence through this Himalaya mountain range. And a study identifies indicator plant species, including 9 tree species, 12 shrub species, and 3 herb species. In our article that is in preparing now, the co-authors make proposal that such species that are significant for each forest type could be used for eco-restoration. Okay, and the most significant impact on species composition and forest type have altitude, slope, and grazing. These 3 environmental variables, grazing is in fact a biological factor, have significant value on our canonical analysis. And thank you for attention.
Thank you very much for very interesting presentation and time discipline. Thank you very much. And now I invite Ms. Agnieszka Hutniczak, who will talk about variation of cell wall chemical composition in the leaves of silver birch in different habitats in urban and industrial landscape.
Good afternoon, ladies and gentlemen. I would like to welcome you. It's great that so many of you were able to make it this morning. So let me introduce myself. My name is Agnieszka Hutniczak and I am from Silesian University in Katowice. At the beginning, I would like to thank the organizers who allowed me to give the presentation. It gives me enormous pleasure, indeed. The presentation is connected with my research interest and it is under the title Variation of Cell Wall Chemical Composition in the Leaves of Silver Birch Betula pendula growing in different habitats in the urban industrial landscape. So let me give a speech outlining the most important issue. Ladies and gentlemen, as we all know, the rapid development of civilization, including urbanization and the constant demand for natural resources, has led to the transformation of the environment and the exploitation of the natural capital and consequently to ecological threats, biodiversity losses and climate changes. According to reports, among others Millennium Ecosystem Assessment, we can say that significant threats are visible not only in Europe, but also on a global scale. So changed habitats, no longer resembling natural and semi-natural habitats, can be very important and they are adapted and growing by different plant species, which can adapt to difficult habitat conditions, what we can see also in pictures. The examples of novel ecosystems can be the following. Different post-industrial sites which creates unique specific structure which can be also colonized by various plant species. So now we have got novel ecosystems. In relation to this type of disturbed habitats, it is extremely important to know the adaptability of species to extreme conditions. For this purpose, various research methods, including landscape, supraorganismal, organismal, and molecular levels of research, should be carried out. Interdisciplinary research, which provide insight into understanding of entire ecosystem functioning, are valuable important. Due to the fact that the chemical composition of cell walls of plant leaves in relation to the environment conditions is not sufficiently known, an analysis of these issues has been undertaken. So, the use of immunohistochemical tests turned out to be promising in the study in relation to plant cell walls and its changes in relation to different levels of disturbances. So using this technique, we are able and we can explore the localization of pectin epitopes and arabinogalactan proteins in plant cell walls. Pectins are one of the most important elements of plant cell walls, and they play important roles in modulating cell wall structure in response to different stresses and in control of plant growth and development. The most important functions of pectins are presented in the box in the table. The material was collected in 2022 growing season randomly from spontaneously formed patches dominated by studied species. The studied area are marked on the maps, and photos illustrate the studied types of habitats. The prerequisite of this study was the assumption of the different nature of biological processes on 3 types of sites taken into account. So the aim of the presented study was the following: to compare the chemical composition of the cell wall of Betula pendula leaves, including pectins, derived from individuals growing in post-mining habitats, coal heap and quarry, and a control site mixed forest. So the immunohistochemical technique is a laboratory technique which is used to visualize the specific antigen in cells or tissues by using primary antibody. In this slide, I want to present the most important stages of immunohistochemical methods, covering fixation, deaeration, dehydration, and embedding the plant material in accordance with generally accepted method, working with a microtome, incubation of slides with primary and secondary antibodies. The last stage was visualization of immunohistological staining and the light of microscopy level. So we can say that it is a long procedure. Now, I would like to show the results. Firstly, I would like to concentrate on differences in the localization of the arabinan-rich pectin epitope in Betula pendula leaf cells occurring in different types of habitats. In picture A, we can see leaves collected from control site. The signal was present in almost all tissues. In picture B, we can see leaves collected from quarry. The signal was visible in walls of some spongy parenchyma cells. And the last picture refers to leaves collected from hips, and the signal was visible in phloem, collenchyma, and spongy parenchyma. Now, I would like to show the differences in the localization of low estrified pectins in Betula pendula leaf cells found in different types of habitats. So, in picture A, we can see leaves collected from control site. The signal was visible in upper and lower epidermis, vascular bundle, spongy parenchyma, and perifascicular sheet. In the picture B, we can see leaves collected from quarry. The signal was visible in almost all tissue without xylem. And the last picture shows leaves found on hips, and the signal was visible also in almost all tissue, but without parenchyma. In order to make it easier to understand the immunohistochemical results, I want to show the summary of the immunohistochemical analysis in the table, where number 1 means the presence of the signal and number 0 its absence. Moreover, based on numerous photos taken under a fluorescence microscope, we can say that the leaf blade histology was typical for trees, but we come to other surprising results. Differences in the thickness of the leaf blade were observed in individuals from different locations. So, this prompted us to make further observations and explore this topic. So, the second stage was to analyze the size of the leaf blade on cross-section of a given species. In this slide, we can see the example of the leaf thickness of Betula pendula from Kórnik. In the case of second picture, measurement places both in parenchyma and vascular bundle are marked with white lines. Analysis and measurement of leaf thickness were made using appropriate software. These analyses are still in progress. As mentioned, I only presented the localization of some pectin epitopes, but in future, I would like to expand the analysis of pectin distribution using additional types of antibody, as well as to take into account arabinogalactan proteins. We are also considering expanding the habitat spectrum. So, ladies and gentlemen, summing up, we can say that leaves from individuals overgrowing post-industrial habitats were characterized by the absence of arabinan epitope compared to controls. In turn, low estrophyte homogalacturonans were more abundant in leaves from plant growing in disturbed habitats compared to the control site. So, based on qualitative and quantitative differences in the localization of pectin epitopes, we can carry out the comparative studies of species inhabiting different types of habitats. Undoubtedly, in such a way, we can find mechanisms of plant adaptation to extreme habitat conditions. To understand how plant species adapt and try to survive in disturbed habitats, interdisciplinary research should be carried out covering the above and underground level. Only such an approach can provide comprehensive knowledge of ecosystem functioning and environment. Such results can form new sources of knowledge and they can bridge a gap in science. I would like to thank everyone who helped me during my work, Professor Ewa Kurczyńska, Dr. Justyna Wróbel-Marek, and Dr. Ewa Mazur. I would like to thank the leader of our team, Professor Gabriela Woźniak. In the end, I would like to show the list of selected publications referring to the discussed subject. Finally, I would like to quote the words of a famous researcher, Richard Hobbs. Landscapes are increasingly composed of ecosystems that are altered to different degrees. Decisions on when and how to intervene in warring situations need to be made on the basis of the degree of alteration, likelihood of success, and landscape context. Thank you for your attention, and I kindly ask you for cooperation.
Thank you very much for also very interesting presentation. Now I would like to invite Mihan Benali, who will talk about assessment of conservation planting for an endangered species, Quesus duplessiana. Please, the stage is yours.
So, hello everyone. Before I start, let me introduce myself. My name is Hisham Ben Ali. I'm from Algeria. I know that most of you don't know the country. It's in North Africa, and I'm representing the University of Science and Technology Houari Boumediene and the Laboratory of Ecology and Environment. Before I start, I should thank the organizers of this very important and motivating event, the Institute of Dendrology of Kórnik and the Polish Academy. So the work I'm presenting here today is a part of our master thesis entitled Assessment of Conservation Plantings for Endangered Species Cupressus duprisiana albacami. So from the Himalayas to Poland, and now we are going deep in the Sahara. So fortunately, with the international efforts, Algeria is one of the countries that signed and ratified all the agreements about conserving biodiversity. So in our study, we will concentrate on the conservation of an endangered species endemic to Algeria, the Tassili cypress, or Cupressus duprisiana by its Latin name. And this research is aiming to evaluate the conservation efforts within the national territory of Algeria. So we have to present the species before starting everything. So it was discovered by General Dupraz during one of his expeditions in the Sahara Desert, and it was named after confirming that it was Cupressaceae, a cypress, by Aimé Antoinette Camus, a French botanist in 1926. It's known in its local language as Tarut, the Berber language of North Africa, and it's the only conifer of the Sahara. As Professor Grimm said in 1992, he can reach up to 22 meters of height and 12 meters in circumference. And it's monoecious species for Afro-Mediterranean origins. It exists from the Holocene. And it's classified as endangered by the IUCN. So in these two pictures, we show slight difference between other Cupressaceae and other cypress, especially the Cupressaceae atlantica, the Atlas cypress. It differs within the cone size. It's slightly round than the cypress of the Atlas, and it has the flattening leaves, and the color is more greenish gray than green as the other cypress, and it has a very beautiful aromatic odor. So we jump into the conservation efforts, the interest on conserving the cypresses, the Cupressus duprisiana, and I will call it Tarut or Tassili cypress, so we don't go lost in the names. So we'll call it Tarut. Was started in 1961. The French botanist Kezel, I know all know him, planted 2 cypresses in the botanical garden of the University of Algiers, and the real interest in the conservation of this species began in the 70s, where the government established a new natural park in the Tassili cypress, where from this cypress's origins, and yes, and in 1975, a group of Czech Republic scientists collected some seeds and from the Tassili, the original region of Cyprus, and started growing it in vitro and by sowing. Until the 1981, there was the first expedition for planting these plants. For 1991, they planted like around 70 plants in its natural habitat. The second expedition was in the 90s, and the third is recently in 2020. So as I said, our work was to assess and see how the cypress's status is going in Algeria. Our method was to lead foot by foot step, or a strip cruising, as we call it in English, a strip cruising method. So we had to go to each plantation and see if the cypresses were conducting well in different habitats. So I don't talk too much. This is a small version of the table we used in our research. It has the initial number and current number of many of conservation plantings in Algeria. Unfortunately, we can only find those in Janet, in the Tassili Niger and in Algiers that are still existing. All of the others are unfortunately disappeared. Unfortunately, like those in Ajrar, Bashar, Jelfa, and Tamanrasat, those and actually are a part of the same bioclimate. So in this map, all the conservation plantings I mentioned before in Janet, Bashar, Adrar, Tamanrasat are all situated in the Saharan bioclimate, except those of Algiers. We will need this information for later to see the conduct of these cycles. So I will, for the results, I will just mention these two figures because we had a lot of numbers and a lot of figures we had to show, but it's because of lack of time I chose those two. For the first one, the mean circumference evolution in the 1977 Baraki plantation. It's a plantation in 1977 in Algiers. It shows a great exponential growth. From 1982, the plants went from 11.5 centimeters in circumference till 108.48 centimeters from 1982 and to 2023. It's the same thing for another plantation, same in Algiers, Bainam in 1992. The circumference went from 43 to 47 from 2014 till 2023. But the aim and the most important results in our research is the Tarot expeditions. Why is it Tarot expeditions? Because especially those of 2020 were all the same age and were planted in different bioclimate. Some were planted in the Saharan bioclimate, and the Algiers one are in the subhumid climate. So we can make a real difference and about the behavior of the plants and the assessment. So for those of Algiers, there were the three missions, the 1981 and 90 and the 2020s. For the 1981, there was only one, and it still exists. For the 2020, also 3 and 3. The one of the 1990s was because of lack of documentation. We couldn't find where they planted. It must be dead or taken. We go to the Saharan region. From the 1981 plantations, 50 they were planted and documented as planted, but unfortunately 0 exists today. And from the 1990, they planted 4 in the garden, and 0 exists today. And in 2020, they planted 11. They only 6 exists till today, and I shall mention that all these species, all these plants are issued of in vitro culture, not those like those planted in Algiers in Bainam and Baraki. So I will give some morphometrical numbers here. So for those in Algiers, it measures like 800, and the best one was in the Ama garden. It measures like 8 meters, and with a circumference of 1 meter are 43. And we'd love to compare with another one in Janet, but the 1981 is not existing anymore. So let's compare the 2020. So for T1 is the 2020 is the first one they planted. Its height goes from 300 and 400 centimeters, which means 4 and 3 meters. But those in Janet, in the Saharan ones, it's the same age, issued from the same way and the same, almost same days of plantation. The good one goes to 245 centimeters only. Here are some photos we took in Janet in the Saharan region. Unfortunately, some of the cypress planted are not going well. We should have taken a video to see the T4 here in Janet. It was very fragile. If you touch it, it comes off very easily. The other one also, but also there is a slight difference. I think it doesn't show up here, but the one in the Janet T4 here has a slightly more gray color than the T1, which was perfectly green. These photos are one of the dead, not existing anymore cypress in the region of Janet, of the Sahara. As we can see here, we don't actually know, because of lack of monitoring, we don't know what really caused the death of these species in Janet and why. Because as we all know, the Tassili cypress it's in its natural habitat. So all the plantations were dead. And those in Algiers are in perfect conditions. We are asking now what caused that. So I took these photos, and the only obvious reasons that might be the explanation is the quality of water. As we can see traces of white salt there, and maybe excess of irrigation. The lack of monitoring and the assessment is a very big problem in conservation. So before going to the conclusion, I wanted to say that the major problem here in our research is why the cypress of the Tassili is dying. We know that the species is have an apomixis. So in its natural habitat, it's not regenerating. So to conserve it, we went to the in vitro plantations, in vitro cultures, to try and save this species of extinction. But as we saw here, the species in its natural habitat, it's dying, and in the subhumid region is not. So there is a lot of questions we need and we have to reply for. And to conclude, despite the Tassili individuals, as I wrote here, is the long life expectancy. The survival rate in the south is not considerable at all, but in the north, in the subhumid region, it is quite really good. So we don't actually know if it's the in vitro plants, if it's hybrid, or the acclimatation didn't go well. We have to profound more in the research to aim and respond to these questions. So I think this is all, and thank you so much for the attention.
Thank you very much for your presentation, and now I invite Antonina Magdalena Dubińska, who will she will talk about Robinia pseudoacacia in urban forestry management. The stage is yours.
Hello, my name is Antonia Dubińska, and I will present my research on black locust in urban forestry. Robinia pseudocacea is originally from North America, but in the 17th century it was introduced to Europe. This species has amazing ability to tolerate the difficult environmental conditions such as drought or pollution. So in the past it was used to reforestation and for reclamation of degraded soil. Today, maybe in the smaller scale, but we still planted the species in many European countries. Because of this high tolerance to difficult conditions and ability to quick grow, the species is quick spreading in the new areas. And this led us to the problem that it reached 13th place on the list of the most invasive species in Europe, and 5th place if we consider only plants. Today we talk a bit about the climate changing, and this topic will be still in the background of my research. Because of the climate change, we have in Europe problem with some native species. They are suffering because of the uprising temperature and the drought. So we need to find the solution for this problem. And one of the solutions is to introduce alien species which are more resilient to this new climatic condition. Most of the study that I was searching about the alien species and introducing them into the Europe, they are focusing on the traditional forestry and the protected areas rather than on the urban forestry. So the aim of my study was to increase our understanding of the role of black locust in urban conditions, to expand the knowledge on how the species affects on the environment in these conditions, and to verify if the species is a real threat for forest in Warsaw, where I done my research. So I put these three hypotheses that these areas with these species will differ in the species composition compared to areas with native species Scots pine, that they will favor the appearance of other alien species, and that the species is undesirable species for urban forestry. For my study, I choose two stands in Warsaw. Each of them, they are under the supervision of the same institution which is taking care about the forest inside the Warsaw. They have similar size, similar share of the black locust and the Scots pine, which was the control sites for my research. And they are inside the city agglomeration, so the conditions are quite similar. My research consists of the two parts, which are complement each other. The analysis of 80 publications, the field work, where I made 62 plots in these two stands, following these rules from the right side. Each size have 100 square meters, and I was checking there the abundance of species using then the brown-blacked cover class scale. The results for the frequency of species occurrence show that these two stands are differ. In the Nakole forest, this first stand, it was more species. The biggest difference you can see in the ground layer. There, in the Nakole forest is even 2 times more species than in the Lindiego forest. Let's move to the statistical tests. In many categories, which I show here, I found no statistical important differences. But in the Nakole forest, I found that in the shrub layer, there was less species richness on the research sites with Robinia than on the control and mix sites. In the Lindiego forest, there was a little different situation that in the research plots, I found more species than in the control. And the further analysis showed that in this layer we have even more native species than on the control sites with Scots pine. The analysis of the articles was done in the three directions: the social needs, economical potential, and environmental impact. The research about public awareness on the alien species in urban condition in Europe, they point that the public can have a decisive voice on the management plans. So we should be aware about that. But there is not so many research about this problem, so maybe it's a good direction for further analysis in the future. I want to add that these species can be potentially benefits for recreational areas because of their ornamental proposal. In the springtime, as we have now, we can observe the big, beautiful white cluster of flowers. We can see this even next to the coffee place here at the conference. Moving to the economical potential, this species has greater potential in the plantation than in the urban condition, because from there we can get a bigger income from the timber production and from the not-timber products. The most common not-timber products from the Robinia stands is honey, which is very popular from Romania and Hungary, but in Poland you can also buy Robinia honey in the supermarket. But the species can be in the urban condition beneficial for other services, like cooling the microclimate, cleaning the soil from the pollution. But if we need to eradicate the species, it could be a big cost for our budget, because each method that we know now and we are using are very costly, they are long-term and not always give us this effect that we want to get. Most of the study in the publication, they are focusing on the environmental impact. And most of them, they point the negative effect on the species to the environment. For example, on the biodiversity, they show the tendency of the species to dominant in the stands, to homogenize the habitat. One of the mechanisms of changing the condition by the species is that to change the access of the light to the forest floor and to competition in the young age. But not everywhere the species uses the same type of changing, so we should be aware about that and not generalize of the effect of the species everywhere. So my management recommendation for sites in urban condition with the species is to maintain a mixed forest structure, to plant shade-tolerant species for the next generation of trees, to reduce cutting to absolute minimum, to not allow to access the light to the forest floor, and to not introduce black locust on the new sites where we don't have the species yet. For the conclusion, I want to highlight that I found not significant differences in the species composition in stands with black locust and compared to the Scots pine. That the presence of black locust is not necessarily associated with higher abundance of alien species. And that this species does not require elimination because of that in the Warsaw now, but because the climate change climate is changing, so the influence of the species to the environment can also change in the future, and we should monitor the species in the future. Robinia pseudoacacia is promoting shade-tolerant species like maples, but also alien species like red oak. I was also observing this in the forests. this shaded forest floor in this forest can be a long-term and low-cost method for control the species. Maybe to not eradicate the species completely, but just for control. And for me, the most interesting conclusion for this research was that for improving biodiversity and naturalness of the study sites is not enough to just eradicate the species. We need to think about something more, because these sites are very similar that we have with the stand with Scots pine. Thank you very much for listening. This is all from me.
Thank you very much for your presentation. And so this way we finished the first part of presentations, and now I would like to open the time for questions to the authors of presentations, and the questions and discussions. So maybe we start from the last presentation. Yes, maybe please.
Thank you. Do you hear me? Or
I think we can hear you.
Okay, but we have to use microphones. We are obligated to use.
Okay.
I would like to say that we had a very nice presentation.
Thank you very much.
Like the conceptualization. I have two questions for consideration. First of all, is about the comparison. You were comparing Robinia forest with the Scots pine forest.
Yes.
Were these forests in the same habitat, and what type of habitat it was? Nutrient poor or nutrient rich?
Nutrient rich more than poor, but in the city we have disturbed this forest, so it's not that easy to say, but it was more rich than poor. I compare these two stands, Robinia with Scots pine, because they appear in the same stands. So I don't want to take control sites outside of Warsaw or farther.
Yes, I'm not asking because I have a problem with the design. The issue is that if you compare Robinia on poor sites and Robinia on rich sites, the different response you can get if you have different control. If you compare with the poor site Scots pine and rich site Scots pine, it would be a difference. So I think it would be good also to keep in that mind that the effects of invasive species can be dependent on what you are comparing.
Of course.
And you are comparing with what is co-occurring. So
yes,
in my opinion, this design is very good. What I was surprised that you did not find the effect of Robinia promoting other alien species.
It was also a surprise for me.
Maybe it was because this Scots pine forest had on the rich soil had a more developed shrub layer, and generally the species pool, the total number of species was quite low.
The shrub layer was developed well, and I think also affects on this result that I don't take account the boundaries, the edges, because on the edges it was more alien species. I only put these plots inside stands, and I was aware of the edges.
So it's good. Another problem is out. And the third question, maybe later. It would be more methodological, so we will talk after.
You can ask, I think, if I am here now.
I don't know whether it would be not so long, but the question is, how did you pair the, because you have Robinia and Scots pine in the pair, yes? max 30 meters apart on this slide with the methodology?
No, I just in this stance I was just made the group of these plots in the Robinia and in the pine.
So you did not have a pair?
Not the pairs.
Okay, not the pair. Okay, maybe it would be good not care about normality of distributions, but use generalized mixed effects models or linear mixed effect models, including the forest name, 1 and 2 forest, as random effects, because I've seen you use Kruskal-Wallis test because of the normality assumption. But
yes,
to be honest, in biological sciences, in the forest, we almost never have a ideal normal distribution, so I don't care about this assumption and make a linear mixed effects models, and it would improve your results.
Thank you for advice.
Thank you.
Can you say a little more about the method connected to shading, which should stop the expansion of Robinia? I haven't understand.
What's method that we have to eradicate this species?
Yes, yes.
We have like few methods. I think the most common is mechanical, to just cut this tree, but this species has good ability to just sprout from the roots and from the trunks, so it's just not work. We have some chemical and biological methods, but for example, chemical is very hard to implement inside the cities where we have people inside. We can think about some biological methods. Some research, they give example of fire methods, but it's also very hard to implement in the city. It was research of few methods in the Kampinoski National Park near to Warsaw, and they also show that there is not good solution. They don't found any methods that will be good enough. Sometimes they complement few methods in once, they give better results. Some more questions?
No, okay, thank you very much. So maybe later, but so now let's move to the previous presentation about Cupressus. Any questions?
I've got a few questions to your presentation, Hisham. So, first of all, I was surprised that so few individuals were available for planting. I mean, the species is endangered, but you showed the three expeditions, three efforts, but they resulted in planting just I would say little plants, yeah, a few plants, relatively. So what is the limiting factor for availability of plants? Why is it not regenerating? Is it some seed set limitation, or what's going on?
This is the main issue in the Cupressus dupresiana, because in some projects and some scientific research, they said that the Cupressus dupresiana, especially those in its natural habitat, in Tamrit, in Janat, are attempt of what we call an apomixis. So the species in its natural habitat cannot regenerate. So it's practically almost dead. So what they did, they went to the in vitro culture most of the in vitro culture, if I can remember, was the percentage of how can I say, success. The percentage of success is 0.5%. Also, when we were doing this study, this research, I met with a professor in the Institute of National Forest of Algeria. I asked her about if you saw the 1977 plantations. They are old and big to produce seeds. She said that I did the research and I did not published, but the percentage of success was 4.3%, and they, with the National Institute of Forest, they succeed in producing plants like more than 100 a year, something, but they didn't resist after all. So still now, with the documentation I have, there is no enough research about the Cupressus dupresiana. Some people say that everything has its age, everything must go anyway. But with the Cupressus dupresiana that exists from the Holocene, it survived drought, it survived cold, it survived everything. And in these in this century, you say you have to let it go. It's not right. So we have to do more research to try and conserve this species, especially in its natural habitat. So these vitro plants didn't work. We don't have enough, unfortunately, information about these vitro plants issued by the in vitro culture. If they were hybrid, because we saw in some documentation, especially in French, they tried to hybrid the Cupressus dupresiana, so they can save it ex situ in the sub-humid and the Mediterranean region. But as we saw, it didn't work well. And noting that the 2020 plants are monitored and not well monitored, but they are trying to keep in mind with the conservation of forests there. So that's the main issue.
And you showed also that some of those plantations didn't last. You showed after some re-examination that 0 plants survived. So perhaps you would need more research on cultivation technique of that species also, yes?
Yes,
yeah.
That's we also need more research about the conservation techniques, especially. Yeah.
And do you have the genetic diversity recognized of that species, and I mean the level of genetic diversity within the species, and how does it look like?
Yeah, in my small knowledge, there is no genetically there is no research about this. Most of people, they say there is 2 or 3 sub-families, like there is the Cupressus dupresiana variant, and those that they say that there are some species that were hybrid to the cypress of the Atlas, but there is very few in my knowledge research that shows the real genetic heritage of the Cupressus dupresiana. And this is also an issue if we have to save the species, the real species genetically, or just to save it, or just hybrid it with other species. This is also a very important point, because we have to, I think, we need to save the original species of the extinction. Any other questions? Yes, please.
Yeah, I have a question, because these mature trees grows in the very extreme habitat. And do you have the information or data about the sum of the rainfalls annually? What is the minimum volume of rainfalls in the places that you can found the major trees?
So this is a very hard question because in its natural habitat, it's in the very arid region. So we don't have actually correct and strict data about rain annually. Most of them, they use the main city, but there is like 1,000 meter of altitude difference between the meteorological where the meteorological station and its natural habitat. Some of them they compared it to another region, but it's not with the same altitude. So we don't have actually in the natural habitat in Tassili Mountains, we don't have the exact meteorological data. And also, and to clarify a little bit, those plants in those 3 expeditions, they were monitored. They were not let like that by their own, but still it's an xerophyte species. It can resist to drought and very hot weather, but still with that and with monitoring, they are not existing anymore in the close to the natural habitat region. So this is also another research that we have to do.
So maybe you have some information for how long the tree can survive without rainfalls.
A lot of time.
So this years or?
A lot of years. The last drought that hit the Sahara region, the Tassili Mountains, in the 70s was from 70 or 71 to 89.
Okay, thank you.
10 years of drought. And still with this is another point. How can it resist this drought and not resist the 3 years without rain that we saw these 2 last years?
These are the mature trees which survive, and how about the regeneration?
There is no regeneration. Yeah. But the point is, and also a point that I wanted to, I didn't put a lot of information about in my presentation because it won't be enough 15 minutes. But in one point is the 1977 plantation were issued by grains and seeds originally from the Tassili. So it's, how we can say, it's genetically pure from the Tassili. Those they gave other plants, other small trees, and it's planted in the NRF station in 2011, but we don't have access to the data or to work on it because of the NRF policies. And it succeeded, but they didn't try it in its natural habitat. So hopefully in the next years they will do or we will do something about it. If you have any more questions, please.
So if this tree grows in extremely arid conditions, do you know anything about the mechanisms of how they avoid losing too much water through their foliage? Do they keep their stomata closed most of the time and just avoid photosynthesizing?
Honestly, I don't have such information about
okay, maybe about some other trees growing in similar conditions, or
we don't have data in this. We don't have any trees growing in the same condition. The only only older trees are in the deep in the Sahara, and it's almost like we were the first ones since 2000, I think 2004 or 2003, to go check on those trees. So this is also a very big problem concerning this endemic and ecologically and socially important species. Hopefully next year, starting this, our assessment, others or us, we will start having data. Yes, please, take your time.
the problem with those extreme habitats is that the regeneration events may happen when some favorable environmental window appears. And I mean, maybe there are some conditions which should be met for the species to start regenerating and to set seeds and release seeds, and they could germinate and recruit. And the problem is that we don't know those conditions, and those conditions may not be there anymore because of climate change. And if we don't know these conditions, it would be hard to provide these conditions for the recovery program. You know, because we may try, or you may try to simulate those conditions, but if you don't have this information, it will be hard. Yeah.
It is very hard. It is because also, as I said, the cypresses it survived many of hard conditions. Why did they survive those old conditions and they are not surviving these conditions? And knowing that it's, I think, almost the same temperature in the Holocene after and before than we are now. So we hope that we will have our answers soon. Hope I was clear answering questions. Thank you. Thank you. Any more questions, please?
Okay, thank you. Thank you
Thank
very
you so
much. So now let's move to the presentation of Agnieszka Niczak. Do you have any questions, comments? Okay. Okay, so maybe I will try. Because you mentioned about specific conditions, like urban conditions and industrial conditions, yeah, and about leaf plants. What about needle trees and for instance, using this method in natural conditions, in extremely dry or for instance at pit box or something like this? In such conditions, how
I have to.
do you think this method will working also in these conditions? For instance,
I have to say that this method, immunohistochemistry, is typical for not maybe geobotany department and geobotany methods, because it is this method is used, for example, in laboratory very often. And after the review of literature, I could learn that we have got different results related to species, but growing in laboratory, not on natural, on semi-natural disturbed habitats. But we want to check and we want to examine that maybe plants which grow on post-industrial sites, maybe they differ from another species. For example, maybe they have different features in plant cell walls. And it is key for us. But as I told, it is difficult because we haven't got literature about this subject.
okay, thank you very much. Any other questions, maybe? No, I can see. So let's come back to Himalaya mountains. Maybe some questions. Any questions? I'm too, because you should generally mention about this increase of biodiversity in such line in the salt and such salt line and which turned into direction of Bangladesh, so something
Yes.
like this. Yes, and maybe do you know, or maybe do you know something about studies how it looks from the northern part? Maybe do you know something, or
In the northern part of Himalaya, we have mostly barren lands because it's very dry area of Tibetan mountains, of Tibetan highlands, and there dominate alpine vegetation, and we have no forests. So most forestry areas we have on the southern slopes of the Himalaya mountains. Anyway, there is a difference between east and west because some regions are better recognized and some are recognized in worst way. For example, the Yunnan in China has extremely biodiversity, which is still not very well recognized. We have one project with colleagues from Vietnam because species of Pterocarya tonkinensis occur also in Yunnan in China, and we was able to find one population of the species very far north from other stands. So for me also it is some clue that there can be some populations of the species that are not discovered yet. So if for one species that is quite characteristic for riparian areas we have no data, it means that generally for this forestry area we have lack of data. Even this quite simple analysis, because it was just collection of some plot in the forest, and in this simple method we was able to find species that was not recognized in this area before. So this is in fact very dense. This Kashmir Valley is very dense, have very dense population. So this is not some area that is very far away from the cities without that you cannot go there easily. Just I think the main problem is the lack of money for the research for this region. So I think still we can find a lot of new species in these places. In the first map, I didn't mention it, but we can see it was collection of also this data from the human observation, and we have very high biodiversity around the big cities. This is of course biased because around big cities we have more observation because we have more people. But it's also some indicator that these places like forests still have a lot of species that are not yet discovered, and we can work on it.
Okay, thank you very much. Okay, thank you.
So maybe you found some surprises there, that species that you don't thought that they will be there, or they were more frequently appear that you expect?
It's difficult to me to answer this question because in fact I'm not an expert in this topic because I'm here as a substitute in fact. Anyway, it is mostly herb species that was new discovered, because tree species are quite well recognized. So in case of woody species we don't have any big surprise. Mm-hmm. But maybe a little bit surprising was that we have some strange regionalization, that it was not, the species was not connected like the same lowland belt from in the south and north. I mean that some species from lowlands, for example in the south, was discovered in the most higher elevation also in the south, but was not discovered in the lowlands in the north. but we don't have any obstacles on the way. So it's just a matter of the distance between these plots. And this was visible on this analysis, phylogenetical analysis. And this was a little bit surprising, that in some cases, the species from the lowlands are more similar to the species from the highlands than to other lowland species. And this can be a little bit surprising, but it's a case of adaptation just, and radiation of species.
And maybe when you plan this research and during doing this research, do you think that your results could be useful for better protection of these areas, or for recreation?
Yes, main idea of this research was not only recognize new species or discover something or even describe the species which occur there, but to find some specific typical species from each forest type which can be used in during future regeneration of this forest that was degraded by human influence or some environmental problems. And we found some species that potentially could be used in this way for protection, for regeneration, for rebuild these forest communities.
Thank you very much.
Okay, thank you very much. Maybe another question? No? Okay, so I would like, in this situation, I would like to thank to all authors of presentations, also to all participants for very interesting presentations and fruitfulness, I think, fruitful and and also very interesting presentation, comments and questions and discussions. So thank you very much for this session for all of you, and well, I invite to the next point of the schedule for today, to the banquet at 19:00. Thank you very much.